particularmente orientado a lo que es OVMs y el tema de edición de genes. Mark Freeman, de USDA, presentó la estrategia de Estados Unidos sobre las secuencias, los datos de secuencias genéticas, la política que está sustentando al país en estos temas y algunos ejemplos puntuales. También describió el sistema global de información y el interés por promover el acceso y el uso de información pública disponible, tanto en el trabajo nacional como internacional, para así fomentar la conservación y el uso sostenible de los recursos. Eso parece que es como fue lo más relevante. También dejando claro, y se enfatizó muchísimo en la importancia de este tema en Estados Unidos, debido a que son los recursos públicos los que se están utilizando en gran medida para generar este tipo de información. Entonces, eso me pareció que es clave. Las presentaciones se cerraron ayer con una, y aquí quiero ya calificar, una muy excelente presentación del Ministerio de Ambiente, de Paola Rojas, quien nos presentó el marco regulatorio sobre acceso a recursos genéticos en Colombia. Fue evidente y creo que fue una muy buena oportunidad para todos el conocer cómo ciertamente el país ha avanzado en estos temas. Hay organización, que de pronto es el punto clave. Se nota que el ministerio en estos temas ya está organizado, tienen unas rutas y unos procedimientos que uno como tercero o como usuario del sistema fácilmente puede acercarse y empezar a conocer la ruta para que uno obtenga su autorización. Entonces, me parece que eso es bien importante y hay que mencionarlo. También el hecho de que para Colombia el tema de las secuencias digitales, todo lo que tiene que ver con las secuencias de ADN, se consideran como parte de la biodiversidad, parte del recurso genético, y en ese sentido, pues, aplica la regulación de acceso que el país tiene. También fue interesante conocer cómo, gracias a esa organización que tiene el ministerio, pues ya se han establecido más de 280 contratos de acceso, algunos, o sea, unos 11, 14 con interés comercial. Entonces, eso demuestra que el sistema está funcionando y que se han tenido avances frente al tema. Eso fue lo que se hizo básicamente en la mañana en lo que tiene que ver con las presentaciones. Posteriormente tuvimos una sesión de taller. Una sesión de taller que me parece que fue supremamente importante porque se presentaron unas preguntas que se generaron como resultado de las discusiones y de las presentaciones que se hicieron durante la mañana. Fueron cuatro preguntas, ya vamos a verlas con un grado más de detalle. Lo importante que me parece a mí de rescatar, muchas veces es más importante la forma que el contenido, y aquí hay algo que me pareció fundamental. Juan Diego mencionaba que desde hace muchos años él había visto como una falta de entendimiento, por llamarlo de alguna manera, entre lo que era como ese sector ambiental y el sector agrícola. Y yo siento que ayer algo que no se esperaba, pero que se dio, fue realmente ese alejamiento se rompió. Vimos en las mesas de trabajo, bueno, la importancia de tener estos grupos variados, donde estuvo presente representantes de agricultura, representantes de ambiente, de otras instituciones, y fue muy interesante ver cómo sí se puede interactuar de manera muy técnica sobre los temas. Eso me parece que es fundamental rescatarlo, porque yo creería que en este momento ya no se puede decir, no se puede hablar de un divorcio entre agricultura y ambiente como se venía hablando desde hace algunos años. O sea, aquí se está demostrando que realmente se pueden tratar los temas desde distintas ópticas cuando el tema involucra temas productivos, temas ambientales, temas de regulación, temas de políticas. Entonces, eso me parece que es importante. Aquí simplemente nosotros teníamos 4 preguntas. Bueno, eran más preguntas, pero asociadas en 4 grandes grupos de temas. El primero, bueno, y aquí ustedes no lo están viendo muy claro, me imagino, es el relacionado con los recursos genéticos en Colombia, donde se estaba preguntando, bueno, cómo se le podía dar valor a ese tipo de recursos. Las respuestas ya las vamos a ver más adelante. También tenemos en el segundo grupo de preguntas asociadas con el sistema de regulación global, el sistema de información global. Entonces también tenemos aportes. El tema de las ventajas y desventajas de los derechos de propiedad intelectual relacionados con el tema de secuencias digitales. Aquí pues hubo muchas ideas, les soy sincero, ahí todavía nos hace falta poder concretar un poquito más toda la información que ustedes entregaron. Y una última, muy rápida, relacionada con la estrategia de comunicación. Aquí ustedes están viendo algunas ideas fuerza detrás de las respuestas a esas preguntas. Entonces, por ejemplo, con respecto a los recursos genéticos en Colombia, que eran cómo debe el país dar valor a las secuencias digitales, cómo lo haría, cómo se garantizaría que el beneficio económico derivado del uso de las secuencias digitales llega a todos los involucrados en la cadena de ABS. Entonces, hay muchos comentarios y algunas de las ideas fuerza. Primero, que el valor se estima de varias maneras, hay un valor monetario, hay un valor no monetario, por ejemplo, en el tema no monetario, aporte a conocimiento, aporte, no sé, cultural, la parte industrial que puede llegar a tener un impacto económico, pero también temas no económicos en temas asociados, por ejemplo, con intercambios de materiales, cosas por el estilo. Otra idea que estuvo detrás de estas respuestas es que de pronto el país puede darle valor y puede darle valor a estas secuencias mediante el uso de bases de información o plataformas de información con metadatos que incluyan caracterización completa, o sea, que cuando se suba una secuencia o algo así, en esa base se sepa claramente no solo la secuencia, sino toda la información asociada a la secuencia, de dónde se obtuvo el recurso para sacar la información, bueno, quién fue el que hizo el trabajo de obtención de secuencia, cuál es el investigador, o sea, que haya como una trazabilidad para que se puedan identificar a todos aquellos que han hecho algún aporte con respecto a la información que está disponible en esa base de información. También un punto que se vio por aquí en las respuestas de ustedes es que el valor de las secuencias per se es diferente al valor de las secuencias cuando se involucran ellas en procesos de desarrollo. O sea, una secuencia como tal, de pronto su valor es simplemente asociado con el trabajo que usted hizo previamente para obtenerla. Pero si no se articula con un proceso de desarrollo, si no se le encuentra una funcionalidad que pueda llevar a procesos de innovación, de pronto ese valor pues, va a ser diferente. Conociendo el recurso, es una de las respuestas que se da para cómo darle valor y cómo garantizar el acceso. Entonces hay que conocer el recurso, pero para conocer ese recurso se requiere de mayor inversión, de incorporación de tecnología y de articulación del recurso en procesos de innovación. Entonces es como una cadena que se estuvo mostrando. También esta se asocia mucho con la de las plataformas. Básicamente, para garantizar el beneficio y para darle valor, hay que contar con mecanismos de trazabilidad de la información. Eso es importante dejarlo claro. Y otro punto que se mencionó es que se le va a dar valor mediante la aplicación de derechos de propiedad intelectual cuando aplique, porque ya vamos a ver en la siguiente respuesta que hay unos temas asociados con esto de la propiedad intelectual. Bueno, con respecto a la segunda pregunta, el sistema de información global, el segundo grupo de preguntas, aquí habían preguntas así muy puntuales, me van a hacer a la izquierda. Pues aquí, o sea, derecha para ustedes. ¿Están ustedes de acuerdo o en desacuerdo con un sistema global de información? Si están de acuerdo o en desacuerdo con acceso a ese sistema global de información mediante un pago por suscripción. Y la otra, en qué se diferenciaría ese sistema de información global del mecanismo de información propuesto para el ABS. Entonces, como una respuesta así general, sí, todos están de acuerdo con la idea de un sistema global de información. En donde ya hay posiciones diferentes es en el tema de que no todos están de acuerdo con el acceso por pago, el acceso por pago de suscripción. Algunos argumentos que se presentan es, mire, pues como que no se ve muy lógico que uno tenga que pagar por acceder a la información proveniente del recurso que uno puso. Otros argumentos que se están presentando es, bueno, de pronto, listo, se podrá tener ese pago de suscripción, pero ¿no será que se podrán evaluar las condiciones preferenciales para tener acceso a esa información, conociendo que, por ejemplo, un país fue el que puso determinado recurso o cosas por el estilo? También esto me pareció importante, que el acceso hay que verlo en doble vía. Una cosa es cuando uno es propietario de ese recurso genético, y otro es el propietario de la tecnología y el desarrollador. Pero la situación también hay que pensarla al contrario. Cuando uno como país, por ejemplo Colombia, dice, bueno, yo aquí lo que voy a acceder es a la información aquí ya sobre un recurso genético que proviene de otro sitio. Entonces, ahí hay temas interesantes. Algo que se dijo, no se dio mucha información con respecto al mecanismo propuesto y las diferencias como tal. Lo único que se demuestra es que tanto el mecanismo de facilitación como ese mecanismo que tenga el sistema global de información tienen que tener reglas claras, ¿sí? para que pueda funcionar. Bueno, aquí rápidamente el de las ventajas y desventajas. Se preguntaba sobre cuáles podrían ser las ventajas y desventajas de los dos sistemas, uno con protección de derechos de propiedad intelectual sobre las secuencias digitales y otro que no tuviera la protección sobre las secuencias digitales. Las respuestas ustedes vieron ahí, son muchas y son variadas. No hemos tenido tiempo de distribuirlas en los 4 grupos posibles de respuestas, pero algo que me parece fundamental y que qué bueno que nos lo mostraron ayer, no recuerdo el nombre, pero fue del Instituto von Humboldt, es la importancia de tener claridad sobre este tema. Por definición normativa, en Colombia, las secuencias de ADN, llámenlas como las queramos llamar, son recurso genético y en consecuencia no se pueden patentar como están en la naturaleza y su protección se da en últimas vía acceso, el acceso que se le dé con el mecanismo ABS, que está claramente identificado aquí ya en el país. El asunto está en que las secuencias donde se demuestre una invención y no un descubrimiento son las que serían objeto de la protección mediante derechos de propiedad intelectual. ¿Okay? Eso me parece que es fundamental. Y en últimas, con esa claridad, ya uno podría empezar a dar respuesta a cuál podría ser el impacto en distintos factores, que es yo sobre el tema de costos, si hay algunas ventajas o desventajas. Entonces, en una revisión muy rápida y con lo que ustedes estuvieron hablando ayer, para cada uno de esos factores uno podría encontrar ventajas y desventajas de los dos sistemas. Entonces, por eso queremos ser como más cuidadosos para poner la información, porque en últimas, esto lo que nos está diciendo es que aquí no hay cosa buena, no hay cosa mala. Son distintas alternativas que se tienen para manejar un tema particular que se vaya presentando. Entonces, yo pienso, y eso sí va como con el espíritu de la reunión que hemos venido teniendo, aquí es interesante, no estamos calificando las cosas, ni descalificándolas. Pareciera que como que nosotros lo que queremos es tomar esas cosas, observarlas, entenderlas y con base en eso empezar a tomar decisiones sin necesidad de ponerle esto es bueno, esto es malo. Y eso me ha gustado dentro del evento. Entonces, aquí nos queda como tarea todavía terminarlo, ir completando con base en sus respuestas. Toda esta información se les va a entregar a ustedes, ya les muestro qué es lo que les vamos a entregar, a compartir, pero ustedes van a quedar con esta información. Bueno, y la cuarta pregunta está relacionada con cuál sería la mejor manera de comunicar el tema de las secuencias digitales al público general. Entonces, aquí tuvo ideas fuerza muy directas. Se propone que mediante el uso de todos estos nuevos medios de comunicación, entonces todo lo que son las redes, aprovechar las redes, no sé, Facebook, el Instagram, todas estas cosas, esa es una idea. La otra es que la estrategia de comunicación sobre estos temas va a ser necesario segmentarla en función de los distintos públicos. Habrá que entregar información al sector académico, otra a los tomadores de decisiones, otra a los influenciadores, otra a los reguladores, en fin. Es clave clarificar y alinear la definición de qué es lo que se quiere. Entonces, si ya, por ejemplo, aquí Colombia está tratando el tema de información de secuencias digitales, bueno, que se exprese claramente y que esa definición sea lo más clara posible y que incorpore las definiciones de otros sistemas. Esta que me pareció fundamental, hay que montar una estrategia que permita comunicar los beneficios y los riesgos de este tema para generar confianza. Y no solamente enfocarse en los riesgos, porque esa es una lección aprendida que se obtuvo de ese tema de los OBMs. Entonces, me parece que esas son como las ideas así, fuerza. Importante, estas preguntas dan para mucha discusión y para más trabajo, pero si somos sinceros, a cada una de estas le dedicaron máximo 25 minutos. En grupos de 12 personas, 13 personas por cada mesa, así que yo siento que el trabajo que se hizo fue más que bueno y ayuda como a motivar la discusión más profunda dentro de sus instituciones o entre instituciones. Entonces, con eso ese era como el objetivo que se tenía de esa actividad. Así que la dimos por cumplida. Con eso yo creo que ya damos por terminado lo que sería esa primera parte del reporte del taller y las conclusiones del día anterior, y ahora vamos a desarrollar nuestro programa del día de hoy, que va a estar enfocado en biología sintética y edición de genomas. Vamos a tener presentaciones, así como al día de ayer. Entonces hay una sesión de presentaciones y posteriormente, ya en la tarde, vamos a tener una presentación, también unas presentaciones, y luego cerramos con un taller. Esa es en principio la dinámica. Entonces yo creo que ya sin más podemos darle, entonces ya le paso si quiere ya. Ya está, entonces si quiere lo.
Entonces ya.
Perfecto.
Bueno, muy buenos días a todos, muchas gracias por continuar el día de hoy. Vamos a darle la bienvenida al doctor Daniel Boitas. Ya se está conectando. El doctor Boitas es profesor del Departamento de Genética de la Universidad de Minnesota. Actualmente es director y cofundador de la compañía Calyx. Él es PhD de la Escuela de Medicina de Harvard y descubrió y desarrolló una poderosa tecnología de edición de genomas que se conoce como los Talents, o las nucleasas efectoras similares a los activadores de transcripción, y actualmente está desarrollando modificaciones dirigidas y eficientes en genomas de diferentes variedades de plantas para avanzar en biología básica y desarrollar nuevas variedades de cultivos. Además de ser profesor de la Universidad de Minnesota, como mencionaba, el doctor Boitas cofundó Calyx. Entonces, le damos la bienvenida al profesor Boitas. Daniel, could you hear us?
Yes, I can. Can you hear
Great.
me okay?
Yes, we can hear you. So
Great.
you can start.
Thank you very much.
Thank you very much.
I really appreciate the opportunity to speak today. I apologize I can't be there. As you can see, I'm a professor and I also work at my company, but today's the first day of the new semester. So when I finish my presentation, I have to give a genetics lecture to 100 students. So I feel very fortunate that I can work both at the university and at a company because I've been able to develop technology at the university and optimize it and then see its application in the real world. So today's presentation, I'm going to draw from both my work at the university and my company, telling you about the technology, gene editing technology, how it works, and how we've begun to commercialize it. In fact, just this year, we've begun selling the first food ingredient that was created through gene editing. So gene editing is really a process of DNA repair. It was in the 1990s that we learned a considerable amount about how DNA damage gets repaired by cells. And in gene editing, we've just harnessed this knowledge so that we can direct repair outcomes to edit the genetic material. So in this slide at the top, we have an image of a chromosome, and the type of damage we create is a double-strand break. We actually sever the chromosome. And then the cell, because it wants to protect its DNA, it repairs it in one of two ways. It either rejoins it through non-homologous end joining. Sometimes errors occur, and so you create mutations or alterations to the DNA sequence. The other pathway of repair is called homologous recombination. And here the broken chromosome copies information from a template. And so we can provide to the cell this template, and we can really copy any information into the genome we wish. Single base changes, large DNA insertions. This is a really powerful application of the technology and a powerful way to make precise changes to genomes. So the question in the field for many years was, how do you go about making that targeted double-strand break? And this slide shows the reagents that have been developed over the years. Meganucleases, these are proteins that naturally bind and cleave DNA, and efforts were made to engineer them to specific and new targets in a genome, but they've been very difficult to work with. When I began in the field, I was working on zinc finger nucleases. These are proteins, these red spheres each represent a protein that binds 3 bases of DNA, and then it's fused to a nuclease to make that targeted break. My contribution to the field were the talons, and I'll talk a little bit more about how these work. But again, these are proteins that recognize DNA and introduce that double-strand break. So the talons are derived from plant pathogens, actually. So this is a slide of a plant cell that's being infected by a bacterium of the genus Xanthomonas. And so this bacterium delivers these proteins called tal effectors to the cell, and they bind a DNA target. And once they bind that target, they have a transcriptional activation domain. They can turn on this gene. They can increase the expression of this gene. And these genes actually benefit the bacteria. They make and export sugars, for example, that feed the colonizing bacteria. So one of my colleagues, I was a professor at Iowa State, one of my colleagues, Adam Bogdanov, discovered how these proteins bind their DNA targets. And that's shown in this slide. Similar work was also carried out in Ulla Bonus's lab in Germany. So this is the tal effector. This is the protein that's delivered to the plant cell. And it's this repeat domain that binds to the DNA. So each of these repeats has pretty much the same amino acid sequence, except residues 12 and 13. And it's these residues that determine what base is bound. So if the first repeat has HD at this position, it binds C. The second repeat has NG, it binds T. A and G similarly are bound by different amino acids at this position. So it's a very simple and elegant DNA binding code. So this slide shows the structure of that amino acid motif. So these amino acids up here fold into a hairpin, and it's this residues 12 and 13 that reach into DNA and make a base contact. And so the tau protein wraps around DNA. Again, a very elegant mechanism of binding. Each colored domain here is one TAL effector that's binding a specific base in DNA. So my laboratory, we decided to utilize this DNA binding mechanism to create DNA editing reagents. So here are those TAL effector repeats that, in this case, we designed to recognize a specific DNA sequence. So there's one array here, and we fuse them to a nuclease such that when the nuclease comes together, it makes that targeted DNA break. So the TAL effectors prove to be very effective at DNA targeting. We can really target any site in a genome. And then this allows us to introduce the breaks and direct the repair of the broken chromosomes in the manner that we so desire. And I'll be giving some examples of that throughout my presentation. So shortly after the TALENs were discovered, CRISPR-Cas systems were described and understood. And in each of these other cases, it's a protein that binds DNA to create the double-strand break. But CRISPR systems are a nucleic acid, an RNA that pairs with DNA. And then this, in turn, is complexed with an enzyme that then makes the break. So this is an even simpler mechanism of DNA targeting. You don't have to design and engineer proteins to recognize DNA. You can simply do DNA targeting by expressing this RNA and the protein that then makes the break. And so for most laboratories, most academic laboratories work on CRISPR-Cas. The use of the other technology platforms is really determined by intellectual property. So when we developed the TALENs, we filed patents that were then licensed. And similarly, zinc finger nucleases, there's quite a few patents around them. So some companies like Sangamo Biosciences works on zinc finger nucleases. Other companies like Precision Biosciences works on meganucleases. And then the CRISPR-Cas intellectual property has been, a focus of considerable debate, and intellectual property seems to be owned by a number of entities, and more patents are being filed and issued all the time. So the IP around CRISPR-Cas remains complex. So I'd now like to talk a little bit about my experience in the commercialization of the TALEN technology. So as I mentioned, when we discovered the TALENs, we filed patents on their use for engineering genomes. So the University of Minnesota, who owns the intellectual property, licensed that IP to Selectis, a French biotech company. And Selectis is using gene editing with TALENs to engineer the immune system to fight cancer. So they harvest T cells, edit them, and put them back in patients, and then those T cells then recognize cancer and remove it from the body. So Selectis early on recognized that the intellectual property also had applications in plant agriculture, so they formed the subsidiary Calyx, which I became involved in very early on. I was one of the co-founders of the company. And this company is now publicly traded on the NASDAQ stock exchange, and we're advancing, and I'll describe about some of the projects that we're advancing and commercializing. So, who is Calyx? Well, the company has changed over the years, as you can imagine. In the first years, we were focused almost exclusively on implementing the technology and optimizing the technology. But then, of course, you ultimately need to commercialize your technology. And so that necessitated defining the mission of the company. And so our goal is really to develop healthier food ingredients. if you think of agricultural biotech in the past, most of it was focused on farmer needs, creating herbicide tolerance or pest and pathogen resistance. But we really thought there was an opportunity with gene editing to meet consumer needs. And so that became the focus of the company. And more specifically, we wanted to address food-related issues. In the US, obesity has been increasing at an alarming rate. Poor diet contributes to increases in diseases such as diabetes and heart disease. Food allergies are increasingly prevalent. And so what we felt is that we could partner with the food industry to use gene editing to provide food ingredients that the consumers want, that are traceable, that we can understand where they came from, and so that these food ingredients have to go under less processing. And so our goal is to increase healthy food ingredients like dietary fibers or fatty acids that are healthy, and remove undesirable features from food like trans fatty acid, allergens, or other anti-nutritionals. So I'll tell you now a little bit about the first genome-edited crop to enter the food supply, a crop that we've just begun, or a food ingredient that we've just begun to sell this year. So a little bit about how the science behind this first gene-edited food. So we created this product through this mechanism of DNA break repair. So what we did is we created a break this in the soybean genome, so this is a soybean product, and then asked for mutations at the target site. So the way we did it, so this represents the chromosomes, for example, in a plant like soybean. And this is the gene that we want to modify. What we did is we introduced our TALEN construct, that little red box. It can land anywhere in the genome. So we create a transgenic plant with the TALEN, and then that plant, as it grows and develops, it expresses the TALEN. And because the TALEN is very effective at finding its target, it can identify this gene and break the chromosome and then introduce a mutation. And so what we do then is we can cross away, remove the TALEN construct so it's no longer present, and so the nuclease is gone, no foreign DNA remains in the plant, and we've just created the mutation in our target site. So as I mentioned, our first product was in soybean, and of course soybean is a very valuable commodity in the US. It's a number one source of protein for animal feed and the second most important source of oil for human consumption. So soybean oil, however,
What's up? We have, tenemos un problema técnico. ¿Qué pasó? Oh my goodness. ¿Qué pasó?
Se cerró.
En la mejor parte. Bueno, él ya sabe. Oh my goodness. Bueno, vamos a resolverlo prontamente. ¿Qué pasó? You are back. Great. Okay, so we are going to continue. We just have a few minutes to finish. So let's, could you share your screen, please?
Yes. 1 second, please. Let's see.
Sí. Ah, ok. ¿Podemos proyectar la presentación? Okay, perfect. Okay, great.
So can you hear me okay now?
Yes.
Okay. Let's see. I believe I was talking about our soybean product. Is that when we lost
Yes.
connection? Okay. So then I will then just continue there. As I mentioned, our first product, our first gene-edited food ingredient is a soybean product. And what we wanted to do was improve the fatty acid composition of the soybean oil. So normally soybean is very low in monounsaturated fats and high in polyunsaturated fats compared to healthy oils like olive, sunflower, or canola. And because of this, in the past, soybean oil was chemically treated to create more monounsaturated fats. But a disadvantage of this is this chemical treatment called hydrogenation. It produced trans fatty acids, which are not healthy. And in fact, the US government banned trans fats in the food supply in the US. So before banning it, foods were required to be labeled whether or not they had trans fatty acids. And so what this happened is starting in around 2002, with the addition of the label and now the complete ban in trans fatty acids, soybean as a commodity oil decreased over time. Its usage decreased over time. And it was substituted with canola or sunflower or other oils that were naturally higher in monounsaturated fats. So what we did in our gene editing is we wanted to increase the monounsaturated fats. So here's a monounsaturated fatty acid, and it's converted by this enzyme called FAD2 into a polyunsaturated fat. And so we simply got rid of the enzyme through gene editing. We inactivated it, and we increased the fraction of oleic acid and diminished the polyunsaturated fats. So now here's what our fatty acid profile of our oil looks like. It's 80% monounsaturated fats. So it's very close to our target, sunflower and olive oil, in terms of its fatty acid composition. So if you think about the gene-edited product, it doesn't have foreign DNA, as I described. These, in fact, the type of modification is illustrated here. These sequences in bold, that's where our talons bound, and then they made a break in the chromosome in this position. And so rather than adding DNA, we lost a handful of DNA sequences at the target site. It's the loss of this DNA that led to gene inactivation. So we and others who were making gene-edited plant products first sought advice from regulators, importantly the US Department of Agriculture. And these are some of the opinions rendered by the USDA on crop varieties created through this gene inactivation or knockout approach. So as you can see, this is already a few years old, but there are many products that have been created through talons, meganucleases, zinc finger nucleases, or CRISPR-Cas. All of these reagents that can introduce a double-strand break. And pretty much in all of these cases, the genes were inactivated. And so the USDA has been very consistent in their view on gene-edited products created through loss of function mutations or knockouts. And they gave us the go-ahead to plant our soybean out in the field and to test how it behaved. Similarly, so basically, yeah, in the U.S., these products without the foreign DNA were considered non-regulated articles. I'll probably skip some of this regulatory framework because you'll have some discussions this afternoon, I understand, or later this morning. But in addition to the US, we also went to the regulatory authorities in Argentina because we wanted to grow our soybean in the Minnesota winter. We wanted to grow it in the United States winter. We wanted to grow it in South America. And so we also received a regulatory okay in Argentina. And so what this has allowed us to do is then advance our soybean product to commercialization. We're able to grow it in the fields. We're able to expand the seed production and then implement our rather novel business model. So at Calyx, we produce the seed, we contract for seed production, and then we contract with farmers to grow the seed. So the farmers are really our partner. And when they buy our seed, they have to sell it back to us. So here in the United States, it's beginning to be harvest time, and so we'll buy all of this soybean meal or soybean and process it into meal, sell it as protein for animal feed, and then sell the oil as a food ingredient. And so, as I mentioned, we submitted our oil to the Food and Drug Administration for consideration. We provided information on its fatty acid content and amino acid content of the meal. And through this voluntary consultation process, the FDA said that they really had no further questions. And so, as a consequence, we began selling our oil just this past spring. So another advantage of this business model is that we control the grain. So as you know, in the European Union, gene-edited products are regulated as GMOs. And we're still waiting for other countries, particularly in Asia. We're just starting to see regulatory policy being formulated. And so we really only wanted to sell our products in geographies and jurisdictions where we had the regulatory okay. And hence, since we control the grain and its processing and the sale of our products, we control the markets that we deliver our products to. So we never sell or commodity grain. Our grain never enters the commodity chain. So I think I'll now switch and give you another example of how gene editing can create products of value. And in this case, we are editing through this other mechanism. We're breaking chromosomes, but we're copying information into the genome. And so it's a more complex gene edit. But as I mentioned early on, this type of edit can really give you a wide variety of outcomes because you have much more control over the way you change a genome. So this project was funded by the Bill and Melinda Gates Foundation, and it was performed at the University of Minnesota with my students and staff, and also collaborators at the Donald Danforth Plant Science Center. And the goal here was to create a fairly traditional biotech trait, herbicide tolerance. So the Gates Foundation recognized that weed control in cassava is a big problem. And so for the people of sub-Saharan Africa, particularly the women and children, they spend a lot of time out in the fields removing weeds. And even despite this effort, yields are reduced 20, in some cases 80% due to weed infestation. And so the Gates Foundation wanted to increase cassava yields. The motivation for these grants was to increase cassava yields and to get the women and children out of the fields, and particularly so that the children could go to school. That was the humanitarian mission behind the project. So traditionally, herbicide tolerance was created through transgenesis But we didn't want to create a transgenic plant. So rather, we edited a gene that could confer resistance to the herbicide glyphosate or Roundup. And so this is just a schematic of this gene. And what's shown here are mutations that have arisen spontaneously in weedy species that confer resistance to the herbicide. So we wanted to introduce some of these mutations into the cassava genome to create herbicide tolerance. And so before doing so, we created some model plants that were actually transgenic. So we took the native promoter, so this is the gene, and then we introduced those mutations that confer herbicide tolerance, mutations that arose in weeds. We did it under the native promoter and a strong, in this case, viral promoter. And I'll show you some examples of some of those mutations and constructs and the kinds of tolerance that are created. So this slide provides a score of the relative health and well-being of the plant, okay? And so at the bottom we have wild-type untreated plants, so they're just fine. And at the top we have wild-type plants that have been treated with the herbicide. And after by about 10 days, they're pretty much dead. So a score of 7 is dead. And then we have two clusters here of partial tolerance. A gene that has the mutations introduced confers some tolerance, and if you overexpress this gene, you even get better levels of tolerance. And so we were encouraged by this and then went ahead and carried out the gene editing experiment. So this is our target gene, the EPSPS gene. In this case, we used Cas9 to create a break, and we copied in those mutations and a strong promoter sequence. And you can see here are some cells growing on a petri dish with the herbicide. You can see that these are the tolerant cells, which ultimately give rise to small plants. And then we can do green. The hit, they've suffered some. They're not perfectly herbicide tolerant, but still they're very, they're much improved over the wild type plant. And so the question now is, how would these herbicide tolerant plants be regulated? They have a couple mutations in a coding sequence. We changed a promoter. So we really altered that gene, and we didn't inactivate a gene or remove a gene. We replaced and added genetic information. And, as you'll discuss a little bit later today about the emerging regulatory framework, it'll be interesting to see how regulatory authorities like the USDA and governments would consider such types of genome modifications. So it still remains to be seen. And to be honest, even though we've created some pretty nice cassava lines, we've not yet advanced them to field tests or to commercialization. And so, in part, I think because the regulatory, because of regulatory concerns about this product and whether or not it would be considered a traditional GMO or not, because of the types of changes we introduced. So I think the point I'm making is that gene editing can make diverse changes to a genome, and we're now getting regulatory advice and clearance on and understanding on some of the simpler changes that we're introducing. But as the technology advances, more and more sophisticated changes to genomes will be created, such as the herbicide tolerance trait I just described. So I think in the interest of time, I'll skip the last example and just perhaps take some questions from the audience. I'm sorry that I guess we got started late and we had the technical difficulties, but I really wanted, my mission today was kind of introduce gene editing at a high level, and hope that you understand that it's this mechanism of break repair that we harness. I gave two examples, the soybean example where we inactivated a gene to create a trait that's now commercial, and the cassava example we copied information and made more sophisticated alterations to the genome, but still to confer a trait of value and interest. And then this is my lab at the University of Minnesota, who carried out most of the work. And then, of course, the scientific team at Calyx was responsible for the work in soybean.
Daniel, thank you very much. So we are going to open the session for questions. Are there any questions? Preguntas?
Hi, Daniel. Thank you for your presentation. I have just a question related with the time for obtaining these varieties in soybean, with the mutations and the cost related with the production of the seeds. The other question is related if you talked about of licensing the technology to make be able for researching purposes on commercial purposes. How are you planning to leverage this technology? Thank you.
Yeah, so the first part of the question, how long does it take and how expensive is it to make a trait like the one I described in soybean? So the biggest limitation actually is biology. You know, it just takes a time for the soybean plants to grow. So if we start with the plant, we can create an edited plant in about 9 to 12 months. And then we validate that we've created the edit, and then we have to grow the plant and expand the seed. And that takes a long time. So that one plant, 40 seed, and then we plant those 40 plants and capture the seed from that. And so it takes a few years by the time we have enough seed to commercialize. And again, we grow it in different geographies in North and South America so that we can more rapidly expand that seed. And the company announced when it was initiating its IPO that it was about $6 million to create that soybean variety. And that contrasts to published studies for GMO traits, which can be in the hundreds of millions. And I think we could even do it for less than that, because that was our first product, and we made some mistakes along the way, and it was a learning process. So gene editing can really be quite fast and inexpensive. And then your second question was the technology and how it's used at the university. Is that correct? Or intellectual property in the university?
Commercial.
Oh,
Licensing and commercial purposes.
Right. So as I mentioned, there's the different platforms that have been licensed by different companies. So Precision Biosciences uses the meganuclease platform, and they are developing internally plant-based products as well. Sangamo Biosciences has the zinc finger platform, and already many years ago, they licensed that exclusively to Dow AgroSciences. So now that, since Dow has merged with Pioneer as part of Corteva, that intellectual property, the Talons are licensed to the University of Minnesota patents are licensed, as well as I mentioned there was another group that developed technology that we've licensed them at Calyx for use in plants. And we're now beginning to think about sub-licensing and partnering with other companies to give them access to the technology. And then the CRISPR IP has been confusing. There's a large amount of important intellectual property at UC Berkeley and at the Broad Institute at MIT. And I still think, I'm not a patent attorney, but it still seems to me that additional clarity needs to come forward as additional patents are granted. But certainly both institutes and other institutes, like Vilnius University, have IP which needs to be kind of bundled in order to get freedom to operate. And in my opinion, it's still a complex and intellectual property environment around CRISPR.
There is another question here.
Thank you, Dan, for your presentation. I'd like to know what is the origin of this cassava before the editing. Is come from a commercial company or is come from the farmers in Africa? And just this question is just to know if you had to pay some royalties or you had to pay the right to use that cassava or that's my first question.
So the cassava varieties we used were cultivars currently being grown in Africa. So we obtained a handful of cultivars from Nigeria, and we had to test which ones we could manipulate in tissue culture and which ones we could edit. So it is a cultivar that's currently being grown in Africa. And to be honest, the Gates Foundation, there's no ongoing efforts now to commercialize those cassava lines. So they're basically just being propagated in the greenhouses. And so, as I said, I think part of it is waiting for a little bit of clarity in the regulatory environment, and part of it is it's a difficult challenge to deliver the edited plants to Africa to teach farmers how to use the herbicide, and so we don't have support for those kinds of efforts. So I think, in my honest opinion, I think what we've created would be a good cultivar for commercial, a more commercial operation as opposed to a smallholder farmer operation. But again, there's currently no ongoing efforts to commercialize those plants.
And my second question is, are these farmers, they are going to be to pay for, once the cassava will be commercialized, the price for the farmers in Africa is going to be the same for everybody else around the world? Or how is going to be that? And just my last question, just by curiosity, why did you name the company Calyx?
So let's see, the first part of the question is, the intention once we created the herbicide tolerance lines was to work with the Gates Foundation and to work with, governments and mechanisms of germplasm distribution in Africa. So that was the intention. After creating the plants, we envisioned a second phase of the project that would involve field trials, testing, and establishing mechanisms of distribution. But currently, those are not underway. So they're not being funded at this time, those efforts. And then the company name, to be honest, it wasn't my choice because it's so hard to pronounce. But we just solicited different names from the employees. And a calyx, of course, is the leaves that hold together the flower, and the flower petal. And so and then it was modified to have the T at the end. And so it has its origin in the flower part.
Thank you, Dan. My name is Paul Chavarriaga from SIAT in Colombia. Did the U.S. required checking in the soybean lines for off-targets? And if that's the first question. And second, do you do self-pollinations with your transgenic edited plants to get rid of the tDNAs, or you just let them auto-pollinate and continue?
Yeah, so when we submitted our
I'm sorry, Dan. I'm sorry. Do you do back process, actually?
Yeah. Okay, I'll answer your second question first. We typically do a back cross from the primary edited plant to remove the trend. If the plant is heterozygous and self-pollinates, you can also just get segregation from the self-pollinated material, but we typically back cross right away to remove the transgene. And then we do, so for example, when we presented a data package to the Food and Drug Administration on our soybean oil, What we did in terms of off targets is look at, I think it was 10 to 12 most closely related sites in the genome to see if there were mutations in those sites, and there were not. And then we sequenced the genome at, I think it was like 30x coverage to ask, was there foreign DNA that landed in the genome that we weren't aware of? And so we provided that data in our regulatory package. Those were the two kinds of checks for unintended genetic consequences in the plant material.
Are there more questions for Daniel? ¿Más preguntas?
Daniel, I have seen that your company went through an IPO in 2017. What can you tell us, being a scientist, about the process of creating a startup and getting involved with venture capital funds and getting the funding and getting to an IPO? What can you tell us as a scientist about the process?
Well, so I didn't tell you this in my talk, but before forming Calyx, I'd started another company that used the zinc finger nuclease platform for gene editing in plants. And we got it working, we published papers, but we didn't have access to the intellectual property. And the funders of the individuals who founded the company and provided capital, they could not come to a satisfactory terms with the holders of the intellectual property. So that company just ceased to exist. So then I went back to the university full-time, and then we invented the talons that I told you about. This time I filed a patent right away. And I knew I wasn't a businessman, and so when Selectus approached us and said, let's create an ag biotech subsidiary, I said, okay, I'll help out, but I don't have the expertise in financing and intellectual property and regulatory policy, etc. So they provided the resources needed to get the company going, and I could focus mostly on the science. But I think that was an important lesson, that I'm a scientist first and foremost, and less a businessman. And then as the company grew and we needed to raise capital and separate ourselves from the parent, that was the IPO. That was what the IPO was about. And so I spent a lot of time helping. Investors wanted to know the science and understand the science, so I spent a lot of time talking about the science to the investors, about intellectual property, and about the kinds of products that we could make with talons. So I've been involved more as an advisor than a regular employee of the company.
Well, more questions? ¿Más preguntas? No? Okay. So, Daniel, thank you so much for your very wonderful and excellent presentation. Very nice. Thank you.
Thank you. Enjoy the rest of your meeting.
Thank you. Bueno, entonces, continuando con nuestro programa, vamos a seguir con la siguiente presentación, que está a cargo de Chris Peterson. Él va a hablarnos sobre las perspectivas del gobierno de los Estados Unidos en relación a los productos de biotecnología. So Chris is going to do a presentation.
Gracias. Buenos días. That's about the end of my Spanish. Now, to begin my talk, I'm going to ask my colleague to stand and pay close attention. You come up here. Most of you are scientists, so you're trained observers. This is Chris Peterson. This is David Herron. We added this mystery to the program just to keep you awake. We know sometimes the interest on the second day starts to go down. So the mystery is now solved. I am David. This is Chris. Nice to meet you. Mucho gusto. El gusto es mío. Okay. Those are my next two phrases I know. I couldn't ask for a better talk to lead into my presentation. And I noticed that I'm the only regulator on the program. And the question comes back many times with these techniques. Where do these stand in the regulatory system? The PowerPoint presentation I have today is a single PowerPoint, so when you get your copy, you'll realize that Chris is on the program, on the PowerPoint, and I'm on it. I'm a regulator. I'm with the USDA's Animal and Plant Health Inspection Service. Chris is in the Foreign Ag Service, Agriculture Service, so their interest is in selling U.S. agricultural products around the world. My focus is only on safety, so I have the easy job. Some of you I've had a chance to speak with. Perfect. Gracias. Some of you I've had a chance to speak with already, and I've told you my background is as a plant pathologist. And I usually start my talks by a small confession. When I started in 1991 with the Animal and Plant Health Inspection Service, with the Biotechnology Regulatory Program, I was leaving the research bench where I was looking at plant microbe interactions, and I was going to work for a regulatory agency. And I thought it was going to be very boring. But I had a young family, so I tried to be flexible. And I'm not very good at telling the future, because since 1991, I have not had a boring day, not one. It's been a very pleasant surprise. Today, I'm going to talk about the U.S. approach with the regulatory system. Daniel Wojtas's comments, we can talk in the question and answer a little bit more about regulatory definitions. I did not put regulatory definitions in for the EU or for Cartagena or the definition you use here in Colombia. We can talk about that in the question and answers. And I also want to talk in my part of the program, I want to talk about some of the main concepts, the main considerations that regulatory agencies have to keep in mind when they're looking at, especially in this area with genetically engineered organisms, okay? These are lessons that I've learned from talking with regulators around the world over the last almost 30 years. Okay, so we start with a very big universe of agricultural biotechnology for plants, animals, microorganisms. I'm going to focus mostly on the plants. This is where our agency has most of the experience. This is where I think most of you are focused in Colombia and in many countries. But many of the lessons in our regulations and our laws will apply to these other types of organisms. My part of the program is only numbers 1 and 2 on the screen. I'm going to talk about the U.S. regulatory approaches and some of the considerations when choosing a regulatory approach. Those of you who have been involved in the discussions in Colombia about what type of regulations, how you would do this, which ministries, which laws, and all of this, you'll hear some very familiar themes. And I'm going to talk about some of the very practical day-to-day things that regulators look at and almost no one else looks at. Okay? That's in the second part. Chris will talk about, after the coffee break, he'll talk about 3 and 4, international collaboration, and talk about some of the discussions that are taking place under the Convention for Biological Diversity and the Cartagena Protocol. Okay? So I'm only covering 1 and 2. So the U.S. regulatory approach for biotech plants, many of you have heard of this coordinated framework for the regulation of biotechnology in the United States. And this has been described in articles online and in magazines, and it is frequently misdescribed. The Cartagena Protocol is not a law, it's not a regulation. This is not something that was passed by our Congress. It was a policy document, so it's a physical document from 1986, and it's also a concept describing the way that the relevant agencies work together. Okay? So it's not a law, it's not a regulation. The other thing is that it's focused on the safe use of biotechnology. It's not focused on social benefits, it's not focused on consumer right to know. The focus is on safe use, okay? So that's a big, important thing. As this document was being put together in the 1980s, the first draft of it was published in 1983, and it is essentially a catalog of all the laws looking at safe use, looking at laws that USDA implements, FDA implements, EPA, the Environmental Protection Agency, implements, and it's a catalog. That's essentially what most of the document is. And it was looking at the question of whether we already had sufficient laws that we could use for biotechnology. The question was, do we need new laws, or do we already have sufficient laws? The conclusion was the existing laws provided enough coverage. Okay? So there was no need to develop new laws to go to Congress to do that. The other thing is that regulations should be science-based and conducted on a case-by-case basis, and the agencies that are regulating, they should cooperate and coordinate as needed. Okay? That's essentially the document, and the final version was published in 1986. So the first coordinated framework was published in 1986, and then I have to tell you who published it. So this is published not by Congress and not by the agencies. It's published by the White House, by an office called the Office of Science and Technology Policy, and they have periodically come back to these questions. In 1992, they put out a short document updating the coordinated framework, and that document essentially said the agencies should, over time, as they get more experience, streamline their approaches. That was essentially it in 1992. You recognize that 1992, this is when the first genetically engineered plants were getting ready to come out of the research phase toward commercialization. In 2017, there was an update of the coordinated framework looking at more applications of biotechnology. We won't go into that in detail today, but I can provide you with links to that. And then the most recent updating of this was in part of the executive order that again came from the White House just earlier this year, looking at modernizing the approach. Again, looking at the regulatory approach the agencies are using, how well it's working, and also a mechanism for those agencies to report back to the White House periodically to say, how are you doing? Okay? Now, here's a simple example of how the coordination works for one genetically engineered plant. In this example, it's a corn plant, maize, that's been engineered for resistance to an insect that feeds on the corn. The larval stage does the damage, the feeding damage, and they've engineered the Bt gene into the corn. I think most of you in this room are very familiar with this example. In the regulatory responsibilities, USDA-APHIS, my agency, is reviewing for plant health, and I'll talk more about the triggers that each of the agencies have in their regulatory approaches. EPA is looking at the pesticidal substance being produced in the plant cell that allows the plant to resist the feeding damage. And then because the corn is being used for food for humans and animals, there are responsibilities under the Food and Drug Administration for food from plants. Okay? I'll talk a little bit more about the regulatory approach for each of these. Now, the previous talk you heard for the Calyx soybean, Daniel talked about the going to USDA APHIS and asking, does this soybean fall under your regulation? As we look at the triggers used under our regulations, under yours, we always have to go back and read the text and see just what exactly falls under, because anytime you have a regulation, you know that some things will fall under it and some things will fall outside, okay? So, our regulations are supposed to be clear and that anyone can read it and understand it, but in reality, frequently people will read it and they'll say, I'm still confused. And they'll have to go to the relevant agency and say, do I fall under your regulation or not? And this has happened with our regulations since it was first put in place over 30 years ago. In 2011, we started publishing online these communications. In the early years, in the 90s, when I was in the office, someone would write to us, put it in the mailbox, it would come to us through the mail, and we would answer, send back a letter to them. We were the only ones that knew of the communication. Then later, we were doing this by email, and in 2011, we decided, why don't we let others see how this process works, that you can come to us and ask these questions and what the responses are. So we started posting these communications online, and we call this on our website, Am I Regulated? Which sounds very friendly and colloquial, but the person writing is not regulated. And the real question is, does this organism that you have, does this fall under the APHIS regulation? And the thing to remember is the response is only with respect to our regulation. It doesn't affect what obligations they may have under EPA or FDA, okay? That's another thing that is frequently misdescribed in articles online. So, you can write to us. If it meets the definition under our regulation, we don't use the term GMO. GMO is a term that came with European regulations in the 1990s. So under the APHIS regulation, an organism that has responsibility under this particular regulation is called a regulated article. I must admit, when I joined the office in 1991, I thought this was a very strange term. But it's an old term that was used in the plant protection and quarantine part of APHIS, and that's the part of APHIS that originally developed the regulations. So they use this term, regulated article. I'll talk more about our definition, but essentially, to be a regulated article, you have to meet two parts to a trigger. The first part is that you have to have used recombinant DNA techniques, genetic engineering as we call it in the regulation, in making the organism. And the second part of the trigger is that a plant pest has to have been used in making the genetically engineered organism, okay? And this is because our regulations were developed under laws from Congress that had to do with plant pests. So we said, this Calyx soybean actually doesn't meet the definition of a regulated article. You have no responsibility under our APHIS regulation, Part 340, but don't forget you may have obligations to EPA or FDA. Go talk to them, okay? So these letters, and I think we now have about 77, 78 letters, they're all online. And you can see the letter that we received from them, describes the organism, and you can see our response. So, let me come back. There's a very key thing in the FDA description here. Under FDA's authority, all food has to be safe, regardless of technique. So the safety is mandatory, it has to be safe, and it has to be legal. So there's some requirements under the FDA law that say if you've done certain things, then you have to go through certain procedures, okay? I'll talk more about that later. This is the little cartoon that we describe how we coordinate with one another. This is what you frequently see when we talk about GE plants. Now, looking at my agency's definition, under our regulations, you can see we have this two-part trigger. You have to have used recombinant DNA techniques, and a plant pest has to be involved. Either involved as the recipient organism that receives the DNA sequences, the donor organism, or the vector agent. And the vector agent is the organism used to move the sequences into the recipient. So in the early stages, we knew that in 1983, the vector agent that everyone was using was Agrobacterium tumefaciens, the plant pathogen that causes crown gall. So they modified those Agrobacterium strains, removing the plant disease-causing genes and put in whatever genes of interest, okay? So a lot of the genetically engineered plants in the early stages were falling under this definition. The focus when we put this regulation in 1987, when we put it in place, the focus was on having a federal system for doing field testing. There was no provision in the regulation for commercialization. As a matter of fact, one of the public comments that came in said, what is the agency going to do when these new plants are ready for commercialization? And the agency's response was, we'll sort that out later. So you can see, the emphasis was to see, is this a phenomenon that just works in the greenhouse? What happens if you take it to field to regular agricultural conditions? We'll come back to more of the story. Let me see. I'll keep referring to Part 340. There are lots of regulations that APHIS has. This Part 340 is the Biotech regulation that APHIS has. The same law, this Plant Protection Act that our regulations were developed under, this law has regular regulations for plant pests that our Plant Protection and Quarantine implements, and so on. So this one overarching act has biotech regulations, has non-biotech regulations, has regulations for noxious weeds. They're all under this larger umbrella. Okay, here's a little cartoon. For some reason, my brain works better with cartoons. So this is the summary. The regulated article is on the right-hand side of the slide. So it has to be a living organism. That's why the box around it is green. Has to have been genetically engineered. And a plant pest has to be involved. The activities with the regulated article that are covered are importation, interstate movement, not intrastate. So if it's going from California to Iowa, then that's interstate movement. If it's moving from Des Moines to Iowa City in Iowa, there's no obligation under this regulation. And then also environmental release. Now, some of you may ask, what about research that's going on in the laboratory? In the United States, that question was settled in the 1970s, and they decided to use a guideline approach, a guidance approach from the National Institutes of Health, saying here are the procedures you should follow. These are best practices. Your own institution's biosafety committee will make sure that you know how these are to be used. There's no requirement for permits or licenses if you're doing research inside a laboratory, inside a contained facility. Okay? The procedure that we originally had under the regulation, you had to write to us and get a permit. After a few years, we streamlined it and we started a streamlined procedure called a notification procedure. This am I regulated process is what we refer to the process that when we started putting it online in 2011, but it's an old process from the earliest days. People would write to us and say, we've engineered this organism. Does it meet the definition of a regulated article? The link here at the bottom, you can see, go there and see all these incoming letters. Daniel's slide, he had that summary slide, is probably the best summary slide that I've seen yet. And I've seen quite a few, even developed in our office, of the letters to date. His letters went up to 2017. We've had about another 8 to 10 since then. But that's a very good summary slide. Some of the organisms that when we respond, we say they've met the definitions, some have not. Some of the letters have been for organisms that are not even plants. Some have been for microorganisms, some have been for mushrooms, was another example. So if something does not meet the definition of a regulated article, we have no authority under this regulation. So we don't have to make a decision that it's approved. It never fell under the regulation. And this is where I explain to people how regulations work. You actually understand how this works. So imagine you bought an old car and you have a large backyard and you want to drive your car around in the backyard. And you ask, do I need to get a license to drive my car in my backyard? And the law says in almost every country, no, that does not fall under it. If you want to drive it on the public streets and roads, then you need to get a permit. You need to get a license to drive there, okay? So it's not that they're giving you approval to drive in your backyard. That just doesn't fall under those regulations. Same idea, okay? Cars and food. As I said to someone the other day, all my examples, cars and food. Okay, now we go to the Environmental Protection Agency. The Environmental Protection Agency registers all pesticides, regardless, if the pesticides are being used to control ants or mosquitoes or to control weeds. They have to be registered by EPA. The question was, what is the status of when you're engineering a new trait into a plant so that the plant produces a compound and it allows it to resist a pathogen or a pest? And EPA in 2000 finalized their regulation for plant-incorporated protectants. When they first proposed the regulation, they called them plant pesticides. And they had people saying, if you call them plant pesticides, no one will ever buy these because these are going to be used in food. So EPA called them plant-incorporated protectants. But instead of thinking of a pesticide that you buy in a bottle, in this case, a plant-incorporated protectant is the bottle, so to speak, is the plant cell. So the plant-incorporated protectant is the compound being produced that gives the protection from a pest and the DNA sequences that allow that to be produced in the plant cell. Okay? Not all plant-incorporated protectants need to be registered. The plant-incorporated protectants that are there through plant breeding, we know plant breeders and plant pathologists are probably here in the room. Plants produce lots of compounds that allow them to resist pests that are going to eat them. If you develop those plant-incorporated protectants by traditional breeding, there's no requirement for registration. If you develop them by genetic engineering, then you have to get them registered before they're sold. Okay? So EPA's authority is for pesticides being on the market. Okay? Pesticidal substance, this is the definition I just told you. This is the official one. Oh, the other thing that EPA does is they coordinate with FDA and they set the levels that would be safe for pesticide residues to be on food. So they say pesticide levels up to this level, it's safe. FDA enforces that, but EPA does the science to determine what the safe level is. Okay, now we switch to the Food and Drug Administration. Food safety in the United States is a bit confusing. Some types of food fall under one law, some fall under another. all the food from plants falls under FDA authority. Okay? So in 1992, FDA put out a policy statement, and this policy statement still is the bedrock, is the foundation for the FDA approach. And their approach there is not tied to certain techniques. They said, we recognize there will be new varieties, plant varieties being developed. These plant varieties, some of these will be used to produce food for humans and for animals. The food has to be safe, and it has to be legal. In case of this, they said there's no need for a new procedure, so they use a consultation procedure. So this NPV is a new plant variety. They looked at, in this document from 1992, they said these are essentially similar to the foods that we already have. They identified what legal requirements there might be, and then they concluded that the existing legal framework was sufficient and they could just use that. And then they set up a consultation procedure. You can come to FDA, and essentially, as I usually describe it, FDA can check your homework. You would have evaluated whether it's going to be safe, and they review this material. When Daniel was talking about the material that they sent to FDA, and at the end of the FDA consultation, FDA said, we have no further questions. So all of the FDA consultations also are online, and in many ways, those documents provide a more concise and easy to follow description of what the modifications have been to the plants. Okay? So the consultation is voluntary. The safety of any food that you put on the market that's derived from plants is mandatory. You see the distinction. To date, everyone takes advantage of this, because here you have the opportunity to have the FDA experts review all of your safety evaluations. So everyone wants to do that. So there are 164 consultations to date, and here's the link if you want to look at any particular ones. The key question for any regulation now is, to what extent do these organisms fall under the scope? And that depends on the definition of the law or the regulation and on the organism in question. So that's essentially the approach that we're using, that you're using in Colombia, Argentina and Brazil are using, the Europeans have been using. And in the discussion, we can go into more details on that. Now we go to the big picture for any regulators, considerations. Regulatory systems don't occur in a vacuum. They occur in a national context. Usually this national context will include national policies. What are you trying to achieve? That provides the long-range goal, and then the laws or regulations or policies, they're all intended to keep the country moving toward those shared goals. Sometimes there may be incentives that encourage certain choices. Sometimes governments choose, well, we'll just put out guidance. These are best practices. Sometimes the country may decide we need laws and regulations. The distinction here, guidance is not legally binding. Incentives are not legally binding. Laws and regulations are. So laws and regulations have legally binding obligations on the public, and they also actually describe legally binding obligations on the regulators. So the regulations and laws will say what authority you have and how you have to carry it out. And then countries' laws and regulations have to consider international obligations. And in the area for biotech regulation, and Chris will talk more about this, the obligations under the WTO Sanitary and whether it's in biotech or in other areas, they generally try to use this least restrictive approach to be able to achieve the objectives. So this, again, reminds us that you have to be clear in whatever field you're looking at what your objectives are. What are you trying to achieve? So I usually go to a car example here. When you have a driver's license, they give you a driver's license, and if you want to drive to a different city that you've never been to before, imagine if you had a regulatory system that said you need a license, a new license to drive in Cali. You got your license in Bogota, but you've never been to Cali. You don't know the streets there. There's some one-way streets there. You might drive the wrong way. You need to get a new permit to drive in Cali. And then they might say, well, it's the rainy season. You got your permit in Cali in the dry season. The rainy seasons, the roads are very slick. We're very concerned about safety, right? That would be a much safer system. They could check your eyesight every day before you get in the car. So every regulatory system has to make a dividing line, okay? And that's really important. That's something that's very hard for scientists coming into this to recognize. But when I give the driver's license example, people understand. So you have this choice: permits, guidance, and so on. This comes back into play. We had a great discussion at the table yesterday about these new genetic engineering approaches and looking at the big picture. What are the regulatory approaches governments have chosen? National policies, international obligations, safety is what most countries say this is the top priority. Some countries add other areas. They may say we want to have innovation, research and development, harmonization. But usually what comes first is safety to our citizens. Okay, here's a simple cartoon. Different regulatory approaches, no additional requirements from the government. And I have to remind people who are new to regulation that this is actually the most common thing that governments do. No one says you need to get approval before you decide what you have for breakfast. You choose your own breakfast. No additional requirements from the government. You may have guidelines that say a good nutritious breakfast should include these things, not too much of this. Guidance. You don't have to follow it. It's not legally binding. Then you may have a system where there are notification or permits. These are the most restrictive. These set out requirements for how the activity is done. And the expectation from the public is that the government, if they're placing these obligations, the government will verify that people are complying with these. So the question that every regulator has is, can I actually verify compliance? That's not a trivial matter. And we had a short discussion yesterday about what this has been like in the whole area with GMOs, transgenics. Can you verify compliance? So every regulator then has to realize if the regulation says this is a requirement, there's an expectation. The other thing that happens in all of this is that this is a renewing cycle. Constantly. We're in a new phase in the United States. We're re-examining our regulations, how we're implementing this. You're doing the same thing in Colombia. And you're looking at the information we have now, and you're supposed to be constantly re-evaluating. How well is it working? Is it meeting our objectives? Objectives will change over time. The objectives in 1987, as I said, the objective there was have a system to be able to do field testing. We weren't even thinking about commercialization. That could come later, okay? We're in a very different place now. So we have to look at commercialization, we have to look at international harmonization, and so on. So every system is periodically reviewed, evaluated for efficiency, how effective it is it meeting the legal and policy objectives? And then if it doesn't, then you have to go back and sort out the way going forward. And through all of this, you're looking at the most reliable information on how the system is being implemented, both from the science and public policy. Okay? The last two slides that I just showed you are the ones when I say, when I'm teaching classes, I say you only have 2 slides to pay attention to. Those 2. Because I could talk all day off of those 2. But I'm not going to, because this is the end of my part of the program. And now we can open it for, we have time? Plenty of time. Only 5. Okay. And then we have the afternoon. And we have a whiteboard. See.
Thank you. No, just let's. Are you David? David, right? Okay, thank you very much. It was very clarifying. It's difficult to present this, but you did it in a very excellent way. PIPs, plant incorporated protectants. So with gene editing, you can actually have new PIPs that do not produce even a new protein, have nothing, but they do resist pathogens. I'm talking about rice and Xanthomonas, for instance. Are those considered PIPs? Because you can actually mutate a part of the gene, is not the coding sequence even, it's just the promoter to avoid infection. So they are protected. So are those PIPs?
The short answer is that if you use genetic engineering, recombinant DNA techniques, when FDA published its regulation in 2000, they said those, well, they're all PIPs, whether they need to be registered. Mm-hmm. That's the heart of your question. So, and FDA or EPA has been saying that it will provide further guidance on certain types of genetically engineered PIPs, mm-hmm. including if you move a gene, say, from one variety of apple to another variety of apple, it would take too long to do the crosses. So that would be a cisgenic approach. But in the regulation right now, they said, we will re-examine this. They still have not re-examined it. So you would have to go to EPA and ask, say, we use these techniques. Is this sufficient to trigger a requirement for registering this PIP? Okay. So there's no clear answer right now. They've been getting lots of questions for this. Likewise, FDA has been getting the question, if we use these gene editing techniques and make very small changes, can we still come in for this voluntary consultation? And actually, you saw in the Calyx example, Calyx knocked on FDA's door, said, we'd like to do the voluntary consultation. And FDA did not say, go away. They said, okay, we'll sit down. Okay. They're in the process of looking at guidance as we go forward. So it's still a bit of an open question. For my regulations, for the APHIS regulations, one of the questions, some of these are being proposed to be changed. Now, Daniel mentioned that APHIS has proposed a change to its regulation. We published this in early June. We've received comments over 6,000 people. All of the comments, by the way, are online. If you have trouble sleeping at night, you can read these. We are in a period now where we are limited in the discussions that we can have about the proposal. We can basically say, this is what is in the proposal. We can't give any reactions to the comments yet. Because in our system, when we propose, before making a final regulation, the process is supposed to be open so that everyone can hear all the discussions that the agency is having with the public. So if I have discussions with you, only you hear the discussions. So that's my limitation in talking about that. But we are proposing a change to this definition, and actually the term regulated article would go away. No one is crying. So that was the other thing. Daniel said that APHIS decided this is non-GMO. It's not exactly correct because nothing under our regulation is GMO. We don't use that term. Okay? FDA doesn't use that term. EPA doesn't use that term. That's the term that you see in the press and most people will talk about. So that's about where we are now. Other questions?
Thank you, David, for the presentation. I was wondering, what is your take in the off-patent bioengineered products? What do you think? They should be regulated as the same as the transgenic products, or do you think we need a new regulation for that?
For the off-patent ones?
Yeah.
See, this is, for our regulation, our regulation has nothing to do with commercialization. So our regulation and our law is only focused on safety. As a matter of fact, there are some things that are commercially available and they're extracted from plants that still are under the regulation. They have rice where they produce certain enzymes. So they grow the rice, they bring it into the lab, extract the enzymes, and sell the enzymes. We have no authority over enzymes. Okay? So it's a good question for your system here, I know.
David, thank you. David, in your opinion, and maybe not as a APHIS member, but as an outsider, as an expert, just watching the model of Colombia, what do you think, which ones are the major challenges that we need to overcome as a nation to speed up what we are doing right now to characterize and explore our genetic resources and at the end trying to deliver an innovation to society? What is the learning process that we can have as a country based on your model?
Yeah, the two slides that I said are my summary slides. Those are not APHIS slides. Those are mine. And I would say, think of those. And there was a comment that came up in the discussion at our table the other day, actually several times. Any approach has to be practical. When we were talking about the subscription system yesterday, it came up in the discussion, whatever system you have, it has to be practical or it's just not going to work. There are features in the regulatory approaches for genetically engineered plants around the world right now that are not practical. I gave the example yesterday when we were sitting at the table. Your country says if a genetically engineered plant comes into the country, you have to have a permit or a license before it can come in. Well, your inspector is sitting at the port, and they open the box, and they see inside soybean seeds. So they look at the soybean seeds, and they say, I can't tell if these are genetically engineered. I can't tell if these are GMO. So then you have to re-examine, is that a practical approach? The other thing that we won't really talk about much here today, but what comes up in some of the discussions with the public is the regulatory approach that governments choose, they influence how the public sees those activities. If they see the activities as very dangerous, that can be something that they got from the regulations. If the regulations say, you know, we can do it with this level of regulation, then they can see, well, maybe this can be managed by people in a day-to-day approach. I think back to driving our cars, you know. I don't think I answered your question, but it's really those big issues. If the government says you have to do this, the public has an expectation the government will be verifying that, confirming that people are doing it. If you can't actually do that, maybe you shouldn't be putting that in the regulation. So this is part of the process. You know, we said, we learn over time. And every country is going to be different. I mean, many of you have talked with Argentina about their approach and how they do things. That works in Argentina's context. It works under the laws that they have. And then likewise in Colombia, you're going to look at the laws that you have. And we can talk later in the day about this whole thing with definitions, because I think Daniel's talk brought up some very interesting things that when you look at the details of the definitions, and I think Pedro's presentation yesterday touched on this a bit, we can talk more about and some of the things related to the situation in Colombia and others.
Yeah, I have a question about, I know you are involved in regulatory, but what about public perception? How United States or in general, how do you think this is faced and also not stigmatization about GMOs or those kind of things? So how do you see that?
When we first started in the US with the regulations, we had very few people who were concerned. And the early products that were on the market, even the environmental groups said, BT cotton, this is a fantastic thing. Even the environmental groups were saying this because they said, this is a much better approach than spraying the chemical pesticides that were killing birds and other organisms. It was really only until the late 90s, as the discussions in Europe got more excited about saying, well, this was very scary. So in the last 20 years in the United States, we have more people who are saying, this is bad, you shouldn't be doing this at all, you should, you know, our plants are our gift from God, and this whole idea is wrong. So we have that discussion going on in the United States. About 4 or 5 years ago, our Congress had a committee that had a hearing, and they had regulatory agencies come in and they asked this very question. They said, why do we have this misunderstanding in the public about the science, about the safety? What is being done to educate the public? And their conclusion out of that was to give several $1 million to the agencies to develop a public communication outreach in the United States. And that is still, they're working on this still today. Historically, most of our outreach, our public communication from the regulators, has been to the people who have obligations under the regulation. So if you need a permit, we would have on our website, here's how you get a permit. If you want to check the status of your permit, here's how you check the status of a permit. It was very much focused on the people who needed to get permits and get authorizations, get these reviews at the end. Very little focus on the actual education. This is what the science is. This is how it fits into plant breeding. And that actually is because that's a whole different part of the U.S. Department of Agriculture that does public education. And this was one of the things that was frustrating to me. I came from a teaching background, and I asked my first year when I was on the job as a regulator, why don't we tell people the science behind this, explain this? He said, that's not our job. Our job is just to look at the safety. And when I talk to my counterparts in other countries, this is a very common situation, that the regulators sit in one place, and then the government will frequently go back to the regulators and say, why don't people understand? And the regulators sometimes have to say, because our job is to do the safety. You may have a science and technology ministry or an education ministry that's doing education. So the circumstances in every country are different. The approach that we found too is, I'll tell you just a short story. Some of you remember the Terminator technology that was in the 90s. Almost everybody remembers this. When these stories came out in the press, they were all making this sound terrible, and farmers would be enslaved to this technology, and it would be killing our crop. All misinformation got people very scared. And in the Department of Agriculture, they said, this is biotechnology. Who does biotechnology? And they saw, well, the regulators do biotechnology. They didn't even have any system in place. And so we tried in the department, for every story that came out, we tried to then have another story that would say, oh, and here's the correct version. And we realized in very short order, we couldn't keep up. And so this goes back to, I think, the heart of your question is how do you proactively go out and tell people this is what plant breeding is about, the fact, these are the techniques we use all the time. Daniel's presentation today, I thought, was a great way to be able to explain this to policymakers, to the general public. I know when I speak in some countries, they say our outreach goes to the children in the schools because the children go home and they teach their parents. And I thought, that's what works in your country. So it's a big challenge. And frequently, they still come back to the regulators, and they expect the regulators to be able to do it, and we don't really have those tools. Chris will be talking a little bit more about some of this, the public outreach, both domestically and internationally, but it's just something that, it's a difficult challenge.
Yeah. Thank you very much, David. I said it this time right.
Perfect.
Okay.
Muchas gracias.
Gracias. So we are going to a 15-minute break, and then we will come back with Chris. Thank you. Vamos a continuar. Entonces, continuamos con la siguiente presentación por parte de Chris Peterson, de USDA ARS. Chris, welcome. Thank you.
Good morning. Thank you, everybody, for being here. I don't know any more Spanish than Dave does. I studied French, which I keep wanting to reply to you with merci beaucoup, and, you know, but my French doesn't help me very much here, so thank you all for your understanding and the offers of translation and patience. My name is Chris Peterson, and by training, I'm an entomologist and toxicologist. I spent about 20 years conducting pesticide research in a laboratory and got interested in international work a number of years ago. Now, as an entomologist, you can't be an entomologist for the last 30 years without knowing about biotechnology, particularly the Bt varieties and the other areas and the other products that are becoming available. So one of the reasons we were so insistent on presenting these talks in this order is that now that you have an understanding of how the United States structures its regulatory framework, it may be easier for you to understand some of our rationale and goals and priorities as we conduct international collaboration with our partners around the world. And so I'm with an agency within USDA called the Foreign Agricultural Service, and we are a trade agency. We do no regulations, and we don't get directly involved with the research. But where we fill our role is the linkage between the research going on in the Agricultural Research Service as well as the regulation that Dave mentioned, and also looking at how the effects of scientific developments and the regulations affect trade. And so in order for us to conduct trade in agricultural biotechnology, there needs to be a product of science that is being produced. And so we have an interest in the research and development in fostering agricultural biotechnology innovation in the development, commercialization, and science-based oversight of agricultural biotechnology products. This oversight includes coordination with our international trading partners, including Colombia, of course. And conversations on these topics can happen under our participation or the participation of our partners in various instruments, including treaties, international agreements, such as the Wto Sps Agreement, which we'll speak about in a little more detail in the coming slides, various trade agreements, and standard-setting bodies, among others. And so some of the formal mechanisms that are, thank you, Pedro. So some of the formal, there's not only the formal mechanisms that the United States and our trading partners are party to, there are also informal groups and policy and technical discussions that are going on, such as the Global Low-Level Presence Initiative, which I won't go into much more detail on today, and the Like-Minded Group, which is an informal consortium of many countries that share similar regulatory approaches to the United States. Now, one rather recent instrument is the International Statement on Agricultural Applications of Precision Biotechnology. This was proposed in November 2018 by Argentina with 7 other countries on that introduced this on the floor of the World Trade Organization back in November. And what the statement recognizes is that precision biotechnology provides farmers with access to products that increase productivity while preserving environmental sustainability. And it has the purpose of coordinating efforts to ensure that the regulatory approaches for these agricultural biotechnology techniques are based on science rather than other non-science-based factors that occasionally come into the discussion. And the international statement provides the necessary guidelines for preventing regulatory asymmetries, basically preventing a situation where a technology is approved in one country but not in another, and then of course interrupting trade, and of course to minimize any potential trade disruptions due to these asymmetries. So what does that mean for researchers, such as myself in my former life and many of you in this room? Is that with the recognition that precision biotechnology provides farmers with the access to these tools, the scientist's role is that, obviously, to conduct research on and develop effective tools that are going to address the priority needs within the agricultural sector. But there's also a communication role that researchers need to play, which myself included, a lot of times scientists are not the best at. We like to look at a 50 nucleotide sequence and get all excited about a C becoming a G, but being able to tell that to the people who need to know why that matters, a lot of times we're not very good at that. And so that's one area that in my current position, and that came up during Dave's discussion, was that we need to do this role. And that's one of the roles of the scientists going forward on the in all of these discussions, whether it's including such as the WTO SPS Agreement. And so the SPS Agreement, the World Trade Organization Sanitary and Phytosanitary Agreement, came into force in 1995 and is applicable to all WTO members, which currently includes 164 member states. So the SPS Agreement, provides basic trade rules for food safety and animal and plant health standards. The very things that the regulators are regulating, the SPS Agreement addresses. The objective of the SPS Agreement is to preserve WTO members' rights, your individual member states' rights, to provide a level of health protection that they deem appropriate. There's also obligations of the member states to ensure that the rights are not misused and do not result in a disguised barrier to trade. Once again, they need to be based on the science and proportional to the risk that the products pose. And so in doing so, the SPS Agreement sets out the rules by which countries establish their regulatory frameworks for the health of plants, animals, and humans. And these regulatory frameworks, as I mentioned earlier, must be science-based. They must be applied only to the extent necessary for the protection of the health of plants, animals, and humans. And they cannot be arbitrary or discriminatory, and they can't produce any undue delay on the registration and approval of biotechnology products. This applies also to the products of modern biotechnology. And so the SPS Agreement acknowledges that international standard setting bodies such as the Codex, the IPPC, or the OIE. And again, this is where science and scientists and researchers can play a pivotal role in guiding the conversations on the real-world problems, including those problems that introduce an issue for trade. And as mentioned on the previous slides, one of the roles of the scientist is to provide communication to the policymakers so that the policies that are developed don't go against the science that demonstrates how these agricultural products will behave and what risks they may or may not pose. So we'll move on to the discussions under the Convention for Biological Diversity and the Cartagena Protocol on Biosafety. And so, as many of you know, this will be a review for most of you, I'm pretty sure, is that the Convention for Biological Diversity, and it was mentioned on some of the earlier slides, was adopted in 1992 and has been in force since 1993. There are currently 196 parties to this convention, including most of the United States as trading partners. And the convention, or CBD as we say for short, has three objectives: the conservation of biological diversity, the sustainable use of the components of biological diversity, and the fair and equitable sharing of the access of the benefits arising out of the utilization of the genetic resources. And this came up quite a bit yesterday in the discussion on the global sequence data. So, as mentioned earlier as well, there are three protocols under the CBD. There's the Cartagena Protocol on biosafety, which has the supplement to it, the Nagoya Kuala Lumpur Supplementary Protocol on liability and redress. And then there's the Nagoya Protocol on access and benefit sharing, as we discussed some yesterday. Now, the CBD, every 2 years, will hold these large, gigantic meetings called the Conferences of the Parties, or COPs for short. And the purpose of these meetings is to further the implementation of the CBD. And so the most recent one was COP14 in Egypt in November 2018. And the next one will be held in China in October 2020. And I'll go into a little more detail on these in a moment. So, but in between the COPs, there are a number of intersessional activities. It's not something that happens every 2 years and nothing happens in between. There's always some sort of activity going on. And for example, the CBD Secretariat can call for information. There are several calls for information that are open right now on various subjects that countries or individuals or interested groups may be able to provide comment on. Some of those are open only to CBD parties. Others are open to, in a more general sense, to everyone. And these are, more of the, take the form of the public comment periods you may be familiar with, where the organization would provide a statement and upload it. They also have online expert discussion forums, which are more like chat rooms or kind of more unstructured commenting forums that may be able to participate in. There's also something called the ATEGs. That's how we pronounce the acronym, and that stands for Ad Hoc Technical Expert Group. And ATEGs are, they're ad hoc, and so they are established in response to a priority topic that the Secretariat determines. And then the ATEG membership is usually parties only, but not always. There are times where non-parties can take part in the ATEGs. And these ATEGs will meet and then discuss the priority topics and then provide recommendations to the CBD Secretariat based on what was there is also something that we refer to as the SBSTA, which is the subsidiary body for science, technology, and technological advice. I don't know how many variants of the word technology they need in one acronym, but there it is. So, and once again, so the SBSTA, it meets, usually the year of the COP, so it'll meet a few months in advance of the October 2020 COP that we held in China. And during that meeting, they'll frame the issues in preparation for the COP and set the agenda and develop, what's actually going to go on at the larger COP meeting later. So once again, this is an opportunity for scientists to participate in these discussions to help guide the discussions along a science-based track and make sure that people don't, get off into the weeds somewhere and talking about things that are not a priority or not backed up by the best available science. One such issue that is a topic of discussion right now is synthetic biology, which, as those of you who work in the field know, synthetic biology, touches a number of different disciplines, whether that's biology, systems biology, biotechnology, chemistry, engineering. It seems like everybody wants to describe what they're doing as synthetic biology, and with good reason. It's a very diverse field, and that touches a lot of different disciplines. But so as the CBD Secretariat is discussing synthetic biology, it becomes clear that there is no single definition that everybody has agreed on. We talked a lot yesterday about the definition of global sequence data, digital sequence information, and all of those different things, and how everybody seems to have a slightly different definition. So the question becomes, how do we move forward on something when there's no widespread agreement on what that term actually means or what it covers? And so, I believe I turned too many pages. There we go. So the synthetic biology AUTAG has developed an operational definition for synthetic biology. And to quote the operational definition, it's, synthetic biology is a further development in a new dimension of modern biotechnology that combines science, technology, and engineering to facilitate and accelerate the understanding, design, redesign, manufacture, and/or modification of genetic materials, living organisms, and biological systems. Yay, we have an operational definition. But what does that actually mean? So the AUTAG, the operational definition, they recognize that that's a starting point. It's not the end-all definition that is going to precisely define what synthetic biology is, but it's meant to simply facilitate scientific and technical deliberations under the convention and its protocols. And it does not preclude or supersede any national approach. If Columbia decides, for example, to have its own definition of synthetic biology, that's all well and good. The Secretariat is not dictating what the definition ought to be, but it's using it as a starting point, which is a good thing because the operational definition as it is, it's too vague to be useful in a regulatory context. If that definition was adopted as is, it would provide very little guidance on what actually needs to take place. So coming right here in Columbia, we have the the Cartagena Protocol on Biosafety, which was adopted some years ago and has, I believe, about 170 parties, countries that are parties to that protocol. And it governs the transboundary movements and conservation and sustainable use of biological diversity, or transboundary movements of certain living modified organisms. And it's an existing mechanism to discuss the organisms produced from biotechnology with a clearly defined scope of work and negotiated terms. Now, the protocol recognizes that the trade and environment agreements, should be mutually supportive. Basically, that somebody meeting their obligations under Cartagena should not go against other mechanisms that they're party to, such as the Wto Sps Agreement or various other trade agreements or treaties that they are party to. And it also recognizes the need to implement in accordance with obligations under the other international agreements. And one of the points that tends to get lost in discussions on Cartagena is that the Cartagena Protocol, it focuses on a subset of genetically engineered organisms known as living modified organisms, which are defined relatively precisely within the Cartagena Protocol. And one thing that us as the United States want to avoid is people applying the protocol in such a way that it extends the scope of the original meaning by including various elements that may or may not actually have anything to do with the intended scope. So just as there are ongoing discussions under the Convention for Biological Diversity, there are ongoing discussions under the Cartagena Protocol as well. And three such of those are, risk assessment of LMOs, living modified organisms, detection and monitoring in the context of emergency procedures, that's Article 17 of Cartagena, and socioeconomic considerations under Article 26. Now, we could easily spend a couple hours on every single one of these topics. Don't worry, I'm not going to. My plane leaves tomorrow. But this is something that, it's certainly an active area of discussion. And again, another area where the scientists are able to play a role in guiding those discussions along the best available science that the discipline offers. And I was was going to reiterate the same point on this slide that we've already seen, that there are numerous opportunities for government scientists to participate in the various discussions. And so once again, I just wanted to reinforce the point that that research and scientific inquiry plays a very important role in determining how these discussions are shaped, and of course the outcomes and the outputs of those discussions, moving forward. So I'll repeat my some of the few Spanish I know again with muchas gracias. Thank you for your time and attention, and I guess we probably have time for some questions.
¿Hay preguntas para Chris? No questions?
Okay.
No? Thank you very much, Chris. María. ¿Sí? No,
Bueno. Muchas gracias a nuestros anteriores conferencistas y ahora pasamos con el doctor Alberto Rosero. Él es el director de semillas del Instituto Colombiano Agropecuario ICA. Esta dirección tiene a su cargo todo lo que tiene que ver con evaluación de riesgo y normatividad para organismos genéticamente modificados, además de muchísimas otras cosas que hace esta dirección, como permisos, registros, bueno, todo eso que hace una dirección de semillas. Alberto es ingeniero agrónomo de la Universidad Nacional de Colombia y tiene un doctorado en mejoramiento genético de plantas también de la Universidad Nacional de Colombia. Los dejo con él.
Bueno, muy buenos días. María Auxilia, muchas gracias por la invitación para poder participar en este evento. Como dijo María Auxilia, bueno, soy agrónomo egresado de la Universidad Nacional de Colombia. Llevo más de 15 años trabajando en la región técnica de semillas y quien es la entidad, bueno, la dirección responsable de todo el tema de semillas, de semillas.
Habla un poquitico más lejos.
Oh, okay. De semillas convencionales, como modificadas genéticamente y ahorita las editadas. Bueno, entonces vamos a hablar un poco del marco, vamos a hablar del marco normativo que corresponde a todo lo relacionado con la parte de semillas. Perdón. Bueno, yo trabajo en el Instituto Colombiano Agropecuario y ICA, que es la entidad responsable de controlarla y prevenir la sanidad animal y vegetal. Asimismo, es la responsable de controlar la calidad de las semillas y de los insumos que se comercializan en el país. Esta entidad está adscrita al Ministerio de Agricultura y Desarrollo Rural. Bueno, dentro de la parte de semillas, nosotros manejamos un término que es el cultivar. Aquí, cuando hablamos de cultivar, llámese variedad, híbridos, genotipos. En estos híbridos o cultivares podemos tener cultivares obtenidos por mejoramiento convencional o cultivares obtenidos por biotecnología moderna. Y en ese sentido, el ICA tiene un marco regulatorio para cada uno de estos dos mejoramientos. La resolución que nos rige a nosotros es la resolución ICA 3168 del 2015. ¿Sí? Esta es la resolución que controla y reglamenta todo lo relacionado con semillas en el país. Todas aquellas empresas o personas naturales que quieran importar, producir, exportar, hacer investigación o evaluaciones agronómicas, deben estar registradas ante la Ley Técnica de Semillas bajo esta normatividad, la 3168 del 2015. Asimismo, esta norma establece que cualquier cultivar, independientemente si es convencional o obtenido por biotecnología moderna, debe estar inscrito en el Registro Nacional del Cultivar Comercial del ICA. Es decir, si una empresa X, llámese una multinacional o empresa nacional, quiere comercializar semillas de maíz convencional o modificado genéticamente, en una subregión, nosotros manejamos varias subregiones que son Caribe seco, Caribe húmedo, Valle Geográfico Río Cauca, Valle Geográfico Río Magdalena, tienen que montar pruebas de evaluación agronómica para determinar su comportamiento agronómico, mirar cómo es la respuesta a plagas y enfermedades. Estas pruebas de evaluación agronómica deben ser montadas o desarrolladas por una unidad de investigación y fitomejoramiento o una unidad de evaluación agronómica debidamente registrada ante el ICA. Lógicamente tiene que cumplir con unos requisitos, debe contar con profesionales idóneos que tengan conocimiento de las especies que van a evaluar o a mejorar. Como dije anteriormente, estas pruebas se montan en cada una de sus reinos naturales donde se pretende comenzar los diferentes cultivares. Entonces, para esto el ICA tiene establecidos unos protocolos en los cuales estas pruebas se montan bajo un diseño de bloques completos al azar, se montan por cada subregión, se montan unas 4 localidades donde se evalúa el comportamiento agronómico de cada cultivar. Estos cultivares se comparan con un testigo que ya está registrado ante nosotros, ante el ICA. Después de eso, la empresa toma una información, la empresa presenta un informe con los resultados. Entonces se presenta un seminario y de acuerdo a los resultados, el ICA determina si autoriza o no autoriza la inscripción de los cultivares en el Registro Nacional de Cultivares Comerciales del ICA. Una vez el cultivar está inscrito, se puede de este cultivar importar, producir o comercializar semillas. Lógicamente, si quiere importar semillas, la empresa tiene que estar registrada como importador. Si va a producir semillas, la empresa tiene que estar registrada como productor de semillas certificadas o seleccionadas, según sea el caso. Bueno, esta es la reglamentación que aplica básicamente para lo que son los cultivares convencionales. ¿Sí? Esta regulación ya viene vigente desde, esta es la 3168, antes era la 970 del 2010. Ahora bien, con los organismos biomodificados, aparte de la reglamentación anterior, debe cumplir con otro marco normativo, que son la Ley 740 del 2002, por la cual se ratificó el protocolo de Cartagena, el decreto 4525 del 2005, en la cual se establecen las competencias de acuerdo al uso del organismo biomodificado. La resolución ICA 946 del 2006, en la cual se establece aquí el procedimiento de qué es lo que tienen que hacer las personas de las solicitudes para autorización de organismos biomodificados, y la resolución ICA 3168 del 2015, la que vimos anteriormente. En el decreto 4525 del 2005 se establecen 3 comités técnicos nacionales, tres autoridades. Un CTN agrícola, que está conformado por el Ministerio de Agricultura, el Ministerio de Salud, el Ministerio de Ambiente, el director de Colciencias y el gerente del ICA, un delegado. En este CTN Bio, el ICA lleva la secretaría. Está un CTN ambiental, que está conformado por el Ministerio de Ambiente, su delegado, el Ministerio de Protección Social y el director de Colciencias. Y está el CTN Salud, el cual está conformado por el Ministerio de Salud, el director del INVIMA y el director de Colciencias. ¿Cuáles son las funciones de estos comités? Primero, en el caso agrícola, es examinar los documentos de evaluación de riesgo. En este caso aquí, la entidad encargada de hacer las evaluaciones de riesgos para la parte agrícola es el ICA. El ICA, basado en la información que aporta el solicitante, hace una evaluación de riesgo, en la cual la presenta a los integrantes del CTN Bioagrícola. Solicitar información adicional cuando así lo requiera, examinar las medidas de prevención y gestión de los posibles riesgos. Aquí, cuando se autoriza un determinado evento, determinada especie, entonces el CTN Bio recomienda que se tomen ciertas medidas de bioseguridad para el uso seguro de estos organismos aquí en el país. Y recomendar, en este caso al ICA, a la autoridad competente, la expedición del acto administrativo. El CTN Bio aquí recomienda, no obliga. Estos, si bien no es vinculante, el ICA siempre acata estas recomendaciones por parte del CTN Bio. ¿Cuál es el procedimiento que se tiene establecido para que una persona natural o jurídica que está interesada en autorizar organismos bio aquí en Colombia, qué tiene que hacer? Primero, tiene que contar con un registro ante el ICA, tiene que estar registrado ante el ICA, bien sea como una investigación, como un importador o como una evaluación agronómica. Segundo, presenta una solicitud de acuerdo a lo establecido en los anexos 1 y 3 del protocolo de Cartagena, nos aporta una información. Nosotros, como autoridad, verificamos si cumple con todos los requisitos y aporta todos los documentos. Si no aporta todos los documentos, le hacemos requerimientos. Una vez verifiquemos que cumple con todos los documentos y con todos los requisitos, el ICA hace un resumen ejecutivo de la solicitud. ¿Sí? 2 hojas, 3 hojas. Este resumen es publicado en la página web del ICA durante 60 días para recibir comentarios o observaciones. Se reciben los comentarios y si los comentarios son válidos desde el punto de vista técnico-científico, son aceptados. Luego, después de esta consulta pública durante 60 días, el ICA hace una evaluación de riesgos, presenta un documento, se elabora un informe. Este informe se presenta a los integrantes del Comité Técnico Nacional de Bioseguridad y en los cuales, de acuerdo al caso, el CTN Bio puede recomendar hacer estudios de bioseguridad. Así fue como inicialmente las primeras autorizaciones, caso algodón, maíz, rosa, siempre se recomendaron, como no se tenía conocimiento de estos nuevos eventos, se recomendaron hacer estudios de bioseguridad. Estos estudios de bioseguridad se hacen en cada una de sus regiones naturales donde se pretende comercializar. Es decir, si un maíz, un ejemplo, Gilga, lo quiero comercializar en Caribe húmedo, yo tengo que montar estudios de bioseguridad en Caribe húmedo. Si yo quiero ese maíz Gilga, quiero comercializarlo en el Valle Geográfico del Río Cauca, tengo que montar estudios de bioseguridad en el Valle Geográfico del Río Cauca. Eso se hace caso a caso y paso a paso. Y para cada una de las empresas solicitantes. Esos estudios se montan lógicamente con unos diseños, unos protocolos que tienen establecidos, bueno, los cuales recomienda el CTN Bio. Tiene un soporte estadístico bastante fuerte, se hacen en campo. El ICA para estos estudios no tiene establecidos tarifas ni costos. O sea, los costos de estos estudios de seguridad son de responsabilidad de la empresa, pero el ICA es quien toma la información. O sea, el ICA toma toda la información en campo y basado en la información que toma en campo, hace un informe, ¿sí? De acuerdo a los resultados que se encontraron, y esto se presenta nuevamente al CTNBio. Y el CTNBio, de acuerdo a los estudios que presentó el ICA, determina si autoriza o no autoriza el uso de un evento en determinada subregión. Las autorizaciones que el ICA son motivadas, son mediante actos administrativos, resoluciones. Es así como a la fecha se tienen autorizado siembras comerciales, tanto de maíz y algodón genéticamente modificado, lógicamente varios eventos. Estas especies y los eventos pasaron, la gran mayoría por estudios de flujo genético, estudios de seguridad en campo, y de acuerdo a eso entonces ese CTNBio recomendó la autorización de siembras aquí en el país. Estas siembras deben cumplir con unas medidas de bioseguridad, ¿sí? El ICA tiene implementadas estas medidas de bioseguridad mediante actos administrativos. Para el caso del algodón, tiene la resolución ICA 682 del 2009, en la cual hay unos puntos que las empresas tienen que cumplir. Deben realizar capacitación en pretemporada y durante el cultivo sobre el uso de la tecnología. Se debe implementar para las siembras unos esquemas de refugio, son 80-20 y 96-4, y se debe hacer un monitoreo de posibles desarrollos de la resistencia de las plagas que son objetivo. Para el caso del maíz, se tiene establecido también un plan de manejo mediante esta resolución, la 2894 del 2010, en la que aparte de la capacitación se tiene establecido también el refugio 90-10, aparte como es una planta alógama se tiene determinado un aislamiento que no se puede sembrar el maíz en áreas en cultivos no mayores a 300 metros de distancia, y también se colocó por petición de muchos resguardos indígenas que tienen variedades criollas, que estos maíces, estos eventos no se pueden sembrar en resguardos indígenas. A la fecha, estas son las autorizaciones que el ICA ha expedido. Bueno, se han expedido para consumo animal 130 autorizaciones. Aquí podemos ver cómo ha sido la evolución. Para estudios de bioseguridad se han expedido 49 actos administrativos y para siembras comerciales 69. Para consumo animal tenemos especies como canola, remolacha, arroz, maíz, soya. Para el caso de siembras, aparte de algodón, maíz, también se tiene autorizado ornamentales, crisantemos, claveles, gisófilas, rosas modificadas genéticamente. Estos ornamentales se siembran en condiciones de invernadero. En el caso también se tiene autorizado para siembra la soya Roundup Ready, pero la empresa no la comercializó aquí en Colombia. Y estas son las áreas de siembra que se tienen desde que se autorizaron en el 2003. Ustedes pueden ver cómo ha sido la evolución. Inicialmente se arrancó con aprobaciones de algodón en el 2003 y luego en el 2008 fueron las primeras autorizaciones de maíz genéticamente modificado. El algodón, a medida que ha transcurrido el tiempo, ha disminuido significativamente sus áreas, pero no por ser transgénicos, sino por otros costos, por costos del producto, porque no hay subsidios. Y en el maíz se tiene que, durante el año 2018 se sembraron más de 70.000 hectáreas. Bueno, entonces en resumen, la autorización de un organismo modificado debe cumplir con ciertos requisitos. Bueno, primero entonces se hará una evaluación de riesgos y estudios de seguridad si es el caso. Estos, una vez hacen los estudios de seguridad, también se tienen que hacer pruebas de evaluación agronómica para determinar el comportamiento de cada uno de los cultivares. Porque una cosa, nosotros hablamos la tecnología, pero otra cosa son los genotipos, donde está la tecnología. Entonces, se montar pruebas de evaluación agronómica. Que esto conlleva a la inscripción en el Registro Nacional de Cultivares, lo cual lo faculta para importar, producir y comercializar semillas aquí en el país. Bueno, ahora con el uso, bueno, con la aparición de las nuevas tecnologías de CRISPR-Cas, entonces, bueno, hubo la necesidad de hacer mesas de trabajo, un trabajo interinstitucional con diferentes entidades para determinar cómo iba a ser la forma de proceder o la reglamentación que tenía que cumplir estas nuevas tecnologías de editado genéticamente. Entonces, en esta mesa se hicieron unas 4 o 5 mesas de trabajo lideradas por la Dirección Técnica de Semillas, en los cuales participaron delegados del Ministerio de Ambiente, de Agricultura, de Salud, del Instituto von Humboldt, universidades como la Universidad Nacional, el EAFIP, centros de investigación como el CIA, que aquí nos apoyó mucho por el Chavarriega, y el gremio, Acosemillas. Entonces le dije, bueno, mira, bueno, ¿qué vamos a hacer para con esa neutralidad? ¿Cómo voy a hacer la forma de proceder, la reglamentación? Entonces, estos fueron los retos que se plantearon. Revisar y definir qué productos, técnicas o desarrollos no entran dentro del ámbito de regulación de los organismos biomodificados. Otro punto muy claro es no tener ambigüedades en la parte regulatoria, cosa tal que permitiera a los centros de investigación, a universidades, obtener nuevas variedades sin que fueran, bueno, sin que tuvieran trabas por parte del Estado. Y lógicamente estas nuevas variedades irían en áreas de mejorar la competitividad del sector agrícola. Establecer procedimientos formales que estén basados en evidencia científica y que den certeza al sector público o privado para utilizar estas herramientas biotecnológicas. Desarrollar políticas claras y consistentes que le dan a los fitomejoradores lineamientos sobre el marco normativo que tienen que cumplir. Políticas que le den predictibilidad a fitomejoradores sobre tiempos y sobre las inversiones que deben hacer para el desarrollo de estas nuevas variedades. Tener políticas claras que faciliten el comercio y el movimiento de semillas y de productos agrícolas alrededor del mundo, o sea, que como nosotros como Colombia no fuéramos un obstáculo al comercio de semillas entre países. Y se buscó que las políticas a desarrollar por Colombia sean basadas en ciencia y que faciliten la innovación, colaboración y comercio de productos mejorados a través de estos métodos. Y por último, lo más importante, evitar sobre regulaciones al desarrollo de plantas mejoradas. Ya tenemos un marco normativo para la 3168, ya tenemos un marco normativo también para organismos biomodificados. No tenía sentido, no tiene sentido sacar una norma en particular que con cierto requisito, la idea es mirar si estos nuevos cultivares predictados genéticamente, en qué norma los encasillo, si por mejoramiento convencional o los encasillo por organismos biomodificados. Entonces, el término que se maneja dentro de la resolución es innovación en el mejoramiento de plantas, fitomejoramiento. Para el desarrollo de las normas se tuvieron en cuenta antecedentes internacionales de otras normas que han tenido otros países, casos como Argentina, Brasil, Unión Europea. Y también se tuvieron en cuenta las definiciones que están contempladas en el protocolo de Cartagena sobre organismo biomodificado y sobre biotecnología moderna. Y basados en estos, se concluyó que la definición está basada tanto en el proceso como en el producto, porque los productos desarrollados bajo la innovación en el fitomejoramento que no incluyen el transgén caen fuera del ámbito regulatorio. Es decir, si el producto que yo tengo por innovación en fitomejoramiento no contiene un gen foráneo, entonces no lo contemplo como un organismo modificado, sino que lo considero como un organismo convencional. Esta es la norma que salió, que es la 29299. Esta norma estuvo en consulta pública durante 60 días también. O sea, nosotros como ICA tenemos la obligación que cualquier proyecto de norma que se publique pase por consulta pública. Tuvo en consulta pública 60 días, se realizaron los aportes y comentarios, los cuales se tuvieron en cuenta para el desarrollo de la norma. Entonces esta norma la encuentra publicada en la página web del ICA, es por la cual se establece el procedimiento para el trámite ante el ICA de solicitudes de un cultivar mejorado con técnicas de innovación en fitomejoramiento a través de biotecnología moderna, con el fin de determinar si se va Si es un organismo biomodificado y tienen que aplicarle la normativa de ley 740, 4525, 946, o si es un organismo convencional, y se le aplicaría la regla única 3168. Bueno, aquí está el objeto, ya lo dijimos. El ámbito de aplicaciones para todas las personas naturales jurídicas que trabajen con ese tipo de cultivares. Aquí una definición muy importante que se incluye es material genético foráneo y la cual pues, lógicamente se basa el desarrollo de la norma. Es gen o conjunto de genes, secuencias de ADN que forman parte de una construcción genética definida y que han sido introducidos dentro del genoma de un individuo en forma estable a través de técnicas de biotecología moderna, superando las barreras fisiológicas naturales de reproducción. Esta es la definición que nosotros tenemos en cuenta para establecer si un cultivar es un organismo biomodificado o es un organismo convencional. Bueno, aquí, esto es el trámite. Aquí hay unos requisitos que son administrativos documentales. Tiene que aportar una cámara de comercio, tiene que tener un registro ante el ICA, comunidad de investigación o comunidad de investigación o como importador. Y hay unos documentos técnicos, 4 puntos muy importantes. Que eso, primero, cultivo, clasificación taxonómica de la especie. Segundo, cómo fueron las metodologías de fitomejoramiento para obtener la nueva característica. Tercero, si esa nueva característica que obtuvo ya se encuentra presente en la naturaleza y cómo se puede obtener, si la puede obtener de manera convencional. Tercero, las metodologías alternativas para la consecución de esa característica. Y por último, una caracterización molecular que me demuestre que los cambios genéticos presentes en el cultivar en comparación con el material inicial y que muestre la ausencia de material genético foráneo. Entonces, estos son los puntos claves para determinar si un cultivar obtenido por edición genética corresponde a un organismo biomodificado o corresponde a un cultivar convencional. Entonces, en resumen, un cultivar obtenido con técnicas de innovación y fitomejoramiento puede tomar dos rutas. Si determinamos que es un convencional, se le aplica la resolución única 3168 del 2015, en la cual tiene que hacer prueba de acción agronómica, registro nacional de cultivares, lo que le faculta para importar y producir semillas. Si determinamos que el nuevo cultivar corresponde a un organismo biomodificado, debe cumplir con todo este marco normativo. Ley 740, decreto 4525, resolución única 946, en la cual lógicamente eso entra el CTN Bio, se hace la evaluación de riesgo, estudios de bioseguridad si es el caso, pruebas de acción agronómica, registro nacional de cultivares, lo que le faculta para la importación y producción. Bueno, este es el correo para mayor información. Bueno, muchas gracias, no sé si tienen preguntas.
¿Hay preguntas?
Gracias, Alberto, muy buena presentación. Dos preguntas así puntuales. Una, ¿cuánto tiempo está demorando más o menos el proceso? Si el proponente lleva toda la documentación y no hay que devolverle más o menos cuánto toma de tiempo. Y la otra, si han ustedes recibido solicitudes o han dado autorizaciones para materiales editados.
Bueno, para editados se han recibido 2 solicitudes, una para maíz y otra para arroz en investigación. Está en proceso de estudios. Por tiempo, la norma dice que tenemos 30 días para dar respuesta. Si hay que hacer un requerimiento, bueno, o si hay que esperar todo administrativo.
Perfecto, gracias. ¿Más preguntas?
Muchas gracias. No, muy interesante ver que toda la discusión pues en parte ya se ha dado y tienen la regulación en la parte de plantas. Te quería preguntar sobre el aspecto de animales, si se discutió la edición genética en animales.
No, o sea, bueno, yo trabajo en el área de semillas, que bueno, no me compete ese tema. La idea sí, lógicamente, es que en el área pecuaria comienza a tratar este tema que es importante. Igual que en las otras autoridades, tanto como salud como ambiente, también tienen que entrar en ese tipo de reglamentación. Pero en el área pecuaria todavía no se ha empezado a tratar con este tema.
Gracias. Mi pregunta va ligada a la anterior. Yo sé que tú estás en semillas y, bueno, yo trabajo con microorganismos. Tengo no sé si de pronto nos puedes orientar con alguien de ICA, pero ocurre también que, por ejemplo, podríamos estar trabajando con microorganismos genéticamente modificados, pero el fin no es hacer un bioproducto. Es decir, no es para liberación en campo. Entonces, no sé si todos esos pasos tendrá que sucederse, por ejemplo, pruebas de evaluación agronómica no serían, porque este tipo de microorganismos se usarían como para un trabajo diagnóstico. Es decir, se utilizaría un microorganismo que permita revisar o diagnosticar cierta cosa, sea una enfermedad o en mi caso metales pesados. ¿Esos tipos de microorganismos tendrían que tener algún tipo de registro ICA también para poder usarlos y generar un catálogo de servicio con ellos? Gracias.
Sí, en teoría sí deben tener un registro por parte del ICA, ¿no? Lógicamente, pues, no, o sea, en el área que estoy trabajando no le compete esa responsabilidad. Bueno, la parte pecuaria del ICA debe trabajar en un marco normativo para determinar cuál es el procedimiento que debe seguir para este caso, pero en este momento no está establecido.
Una curiosidad sobre la discusión que tuvieron, que tú mostraste que tuvieron en cuenta lo que otros países de la región regularon. ¿Cómo quedó Colombia con respecto pues a la región? ¿O están todos alineados o hay diferencias?
Sí, no, en términos generales son muy similares, se basa sobre el producto como tal. Lo que pasa es que aquí, bueno, hay otras normas que colocan las diferentes tecnologías, bueno, cada una para nosotros no la quisimos incluir aquí, cosa tal que, bueno, que abarcara la gran mayor número de tecnologías que se encuentran presentes para trabajar con editados. Pero en términos generales son muy similares con los países de la región, ¿no? ¿Qué editados pueden ser? OEMs o convencionales. Un OEM, exactamente.
¿Tenemos más preguntas? ¿No? Listo. ¿Hay alguna otra pregunta? Bueno, si no, entonces muchas gracias, Alberto. Seguimos entonces con el panel de las últimas presentaciones. Va a ser moderado por Pedro Rocha. Entonces, por favor.
Ayúdeme aquí con dos sillitas que usted también está aquí. Ok, entonces vamos a tener aquí un panel donde vamos a tener a David, Chris y don Alberto. Y vamos a tratar, si ustedes tienen alguna pregunta, please, you can sit down here, no problem. Si ustedes tienen alguna pregunta que les haya quedado de las presentaciones que se hicieron, si quieren profundizar en algún tema, este es el momento para hacerlo. Y como para ir rompiendo el hielo, a mí me gustaría comenzar con una pregunta puntual dirigida a la final para todos con respecto al tema de OVMs, cómo lo manejan ustedes y después si se puede relacionar con el tema de edición de genes. Consideraciones socioeconómicas. Ustedes, por ejemplo, aquí en Colombia, ya nos explicaron cómo hacen sus análisis para dar la autorización. ¿Ustedes hacen algún tipo de análisis sobre las consideraciones socioeconómicas? ¿Cómo se analiza eso?
No, para el caso de Colombia, las consideraciones socioeconómicas no se tienen en cuenta para las autorizaciones, no hacen parte de la evaluación de riesgo.
Ok, perfecto. I don't know, David, if you can give us some idea regarding socioeconomic considerations and the analysis that you are doing when you are deregulating the products.
So the APHIS regulation is like the situation in Colombia. We look only at the safety. For all pesticide registration in the United States that EPA, the Environmental Protection Agency, does, their statute says they have to look at the risks of using a pesticide and the benefits of using a pesticide. So if that is a socioeconomic consideration, that's something that's built into any pesticide registration that EPA does, and likewise would be a consideration when they're registering plant-incorporated protectants. So that, but that's the same as for any pesticide. I hope that answers the question.
Yeah, okay, thank you. Thank you very much. I don't know if you have... Bueno, no sé, ¿listo?
Yes, thank you very much. I have a question regarding the operational definitions of synthetic biology. If the Convention for Biological Diversity is thinking in somehow to reach a more accurate definition, or how, and if that is the case, how would be that done?
Well, as the United States is not a party to the Convention on Biological Diversity, I'm not sure what role we would play directly in helping to redefine the definition of synthetic biology. So I can't say that I know for sure.
So when Chris was talking earlier about the ad hoc technical expert group for synthetic biology, the U.S. does have a representative who's as part of that group. So all of the ad hoc technical expert groups have representatives nominated from parties, non-parties, and organizations. So it's a diverse group. I can tell you that even to get to that definition that is very broad was a very difficult process for the ATEC. My sense from talking to people is they're not likely to get much farther than that operational definition. This was, they acknowledged that this was as far as they could get to this point to help frame the discussions. But I think your question is a question that others have asked too. And there's no indication now that they're going to be trying to focus more on that. But part of this will depend on what the parties decide in 2020 when they meet. If the parties want the ATEG to refine that definition, it would be up to the parties to ask a group or some other mechanism to do that.
Okay, thank you, David. Aprovechando la respuesta inicial de Chris, relacionada con el hecho de que Estados Unidos no es parte de la Convención del Convenio de Diversidad Biológica, voy a hacer una pregunta que no sé si sea políticamente correcta o no, pero es que a mí me la han hecho. Y es, ¿han considerado ustedes como Estados Unidos ser parte del Convenio de Diversidad Biológica?
Yeah, thank you for the question. I get that question often. So the United States was involved in the development of the Convention on Biological Diversity. We were involved from the beginning through the UNEP. And the United States has, in fact, signed the Convention on Biological Diversity, but our Congress has not yet ratified.
Ok, gracias mil. Thank you very much. It's quite... Bueno, ¿alguna otra pregunta? Comentario. Acordémonos, estamos hoy hablando del tema de edición de genomas, el tema de biología sintética. Tenemos aquí dos países en los que ya se pueden tomar decisiones al respecto. Hemos tenido las presentaciones de algunos productos que han pasado por etapa de investigación, otros que están en etapa comercial. Entonces, con ese marco, por favor, las preguntas que tengamos, no seamos tímidos, no seamos tímidos. A ver, Ricardo.
Si uno produce si tenemos una plaga para un cultivo de importancia económica para el país y se produce esa plaga, se hace una modificación genética, un insecto modificado genéticamente de esa plaga para el control, ¿quién la regula en Colombia? ¿Cuál de esos consejos? ¿El de pecuaria, el de salud o el de plantas?
O sea, si es de uso agrícola, tocaría por parte del ICA, ¿no? De la autoridad. Si es de uso agrícola, pues es una plaga en cultivo, lógicamente tienes competencia del ICA.
Aunque sea animal. Es un insecto, ¿no? El modificado.
Sí, claro. Sí, claro, sería competencia porque aquí lo que se defiende es el uso, ¿no?
Para complementar y podemos poner un ejemplo que yo tuve que trabajar con Alberto, yo soy del INVIMA. Si uno piensa, por ejemplo, un animal como un ratón, pero el ratón está siendo utilizado para investigación en uso de salud. La competencia es del INVIMA, no del ICA, aunque estemos hablando de un animal. Entonces lo que hay que mirar siempre es el uso final de lo que se va a utilizar.
Volvamos a edición de genomas. Y es para Alberto. Estoy descubriendo que de pronto hay un vacío chiquito en la regulación. No, porque es que, lo estaba pensando ahorita, de hecho, y es que cuando usted no sabe si su organismo que creó por edición es clasificado como OVM o como convencional, pues usted no puede ir a campo a hacer una prueba. Entonces, si yo tengo el organismo, así no haya hecho las pruebas en campo y no estoy seguro si funciona en campo porque no lo puedo llevar a campo, tengo que preguntarle a ustedes, primero, que ustedes me definan entonces, no, ese organismo lo consideramos convencional. Ahora sí puedo ir a hacer las pruebas, ¿correcto? Porque generalmente uno cuando se decide a hacer la aplicación es porque tiene los datos.
El producto terminado.
Claro, pero muchas instituciones no tienen la posibilidad de hacer esas pruebas porque no saben si es OVM o no, de acuerdo a la regulación, ¿no?
Me surge una pregunta de lo que tú dices. Según entendí, era que uno tiene que mostrar un análisis genético que muestre que el foráneo no está, ¿cierto?
Sí.
Pero eso, no, o sea, en tu pregunta, no entiendo, cómo es el orden, porque yo pensaría, o sea, después de que modificas, uno podría hacer esa prueba, ¿sí? ¿O no fácilmente?
Bueno, pero si en la parte de investigación de su producto usted trabaja con un OVM, tiene que estar registrado como una unidad de investigación para organismos biomodificados, con la unidad de investigación. Y al final, si el producto, el cultivar ya no es un OVM, lógicamente no le aplicaría la normativa, pero dentro del proceso de investigación tiene que tener usted el registro como unidad de investigación para OVM, porque está utilizando OVMs.
Técnica de OVM, claro. Y la pregunta mía va hacia el procedimiento, porque cuando usted se decide a hacer una aplicación para que le digan si es OVM o no, es porque ya tiene datos suficientes de que el editado genéticamente le sirve para algo, si no, ¿para qué se va a tomar la molestia de hacer semejante vuelta, etcétera? Entonces es como para decidir, hay un limbo chiquito ahí en aquellos que no estén registrados para trabajar con OVMs, por ejemplo, que muchos lo hacen, que muchos no están, ¿no? Muchas empresas no están.
Sí, pero lo ideal es que si usted en su parte de investigación trabaja con OVM, registre su Antélica como unidad de investigación con OVMs.
Ya lo tendría, perfecto. Bueno, ¿alguna otra pregunta? Mientras vamos motivando, una pregunta que no toca a Colombia directamente y que puede tener consecuencias sobre los temas de biotecnología asociados con OVMs, eventualmente con productos de edición génica, con productos de biología sintética. En países con régimen federalista, hemos visto en algunos casos que el gobierno central, o sea, da una directriz y dice esto se va a hacer de esta manera, pero empiezan a surgir algunos departamentos o algunos estados que dicen, no, yo quiero declararme, no sé, libre de transgénicos o libre de este tipo de productos. Con esos antecedentes, ustedes en Estados Unidos como Estado federal, ¿cómo manejan esas situaciones? Si se les presentan casos en los que de pronto un Estado dice, no, yo ante esa decisión técnica sobre un tema de biotecnología asociado con cualquiera de estos productos, OVM o biología sintética o lo que sea, ustedes lo desregularon, yo quiero regularlo. ¿Cómo manejan ustedes esa situación si se les ha presentado?
This is a very profound question. And I see you have studied our Constitution. Because this goes back to the responsibilities in the United States between the federal government and the state governments. So the federal government has certain responsibilities, and any responsibilities that the federal government is not already laid out then the states can occupy. But if the federal government is sort of occupying the field in an area, then that takes precedence over states. So, and that was built into the constitutional system. For example, if, and actually this case came up with the labeling law that went into place. One of the United States, Vermont, wanted to put in place a law for labeling the content of food that might have been from bioengineered organisms. And our Congress at the federal level decided to pass a law occupying that same field, so to speak, in that same area, regarding bioengineered content in food. And as soon as they passed that law, the term they use is preemption. It preempted any state laws that would be put forward in the same area. So that's a mechanism, that's a feature of the relationship between the federal and the states for all areas, not just biotech.
Okay, thank you. Uh-huh.
En español, in English.
Okay.
What was it the case for glyphosate as well? Because in the intent of California to label glyphosate as a possible cancer-producing agent. La pregunta es si fue el mismo caso para glifosato en el California, donde California quiso marcar al glifosato como un posible agente cancerígeno y el gobierno federal le dijo no. Is that correct?
So you've just run to the limits of my legal understanding. As you heard, I'm a plant pathologist by training and a regulator by hobby for 30 years. And the relationship for all pesticide regulation in the United States between EPA and the states does allow for the states, if they have scientific reasons, they can go above. They can also, the other area that you may hear in the United States is for the air pollution standards coming out of automobile exhausts. And so California, because the problems that they had with bad air pollution in the 60s and 70s, they raised the requirements for to have better air pollution coming out of the cars, essentially. And because California is such a big part of the automobile market, all of the automobile manufacturers said, we'll meet the California standard, not the national standard that EPA sets for clean air. So the pesticide situation has some similarities, but Chris actually, his background is in pesticide regulation, so he can give the details on this.
So I missed the names. It was for you actually.
No, it's fine. Well, so a couple points that might help clarify is that, with the labeling example that Dave used, because the federal government passed a labeling standard, that preempts the states. Now with glyphosate, because the federal level has not passed a labeling on glyphosate, there's no federal law to preempt the states from doing that. And in general, a state can be more restrictive than the federal law, but it cannot be less restrictive. So for example, like the legal drinking age for alcohol. If the federal government says that it cannot be below 21, no state can make it 18. But if the federal government says it can be 18, a state can say, well, no, we're going to have it at 21. So they can be more restrictive but not less. Same thing with pesticide registrations. There are certain pesticides that are banned in certain states, although the federal EPA allows them to be used elsewhere. So.
Ok, interesante. ¿Alguna otra pregunta? ¿Comentario? Por favor.
Bueno, pues yo sé pues que está Alberto ahí, pues no es su competencia, pero pues queda en el aire esta consideración, y es que así como los animales, estoy hablando de edición genómica, animales, pues la pregunta que hizo alguien allá atrás, pues no es de tu competencia, es del ICA eventualmente, etcétera, etcétera. Igual queda todo para consumo humano, porque pues es decir, claro, por el sistema colombiano ustedes como ICA regularon las planticas, pero nos quedan nosotros vacíos, entonces quedamos con una regulación hasta cierto punto que no satisface lo que se está viniendo ya digamos en desarrollos. Por ejemplo, me voy a inventar, si llegase aquí el caso de esta mañana que se discutió y es para consumo humano, ¿qué va a hacer el sistema colombiano? Entonces, como más para dejarlo, el gobierno colombiano, ¿qué haría, no? Es dejarlo en el aire. Sé que tú no tienes la respuesta, pero eso como que quedamos como que hemos avanzado, somos líderes en la región, sabemos que Colombia es líder en regulación, etcétera, etcétera, pero desafortunadamente no existen los mecanismos para que una vez, digamos, el ICA tomó decisión, salen los demás y dicen, bueno, yo hago, no hago, me pronuncio, pero quedamos como en ese, sí, vacío.
Sí, de acuerdo. Sabemos las otras entidades, bueno, y tanto también el ICA en la parte pecuaria también debe tramitar su marco normativo para los editados, lo mismo que en el caso de salud, ¿no? De acuerdo.
Un comentario sobre eso, y es que sí, es importante y es urgente que las regulaciones sobre el uso de edición en la parte animal y humana, y sobre todo de medicinas, ¿sí?, sea considerada rápidamente, porque allí es donde realmente el peso de la inversión va a estar, y está. Y además, el primer caso tal vez publicado ya de organismo sintético, biológicamente hablando, con tecnología actual, salió publicado esta semana o la semana pasada en Science. Así que la posibilidad no solo de edición de genomas, sino para organismos vivos, ¿sí?, no plantas, sino otros microorganismos también, y biología sintética aplicada a microorganismos, ya se abrió. O sea que póngase a pensar que, todos pongámonos a pensar que sí es necesaria y urgente una regulación.
De acuerdo.
No sé si hay algún comentario por parte de ustedes. ¿Do you have any comment? No. Bueno, Gabriel. Y con esta creo que vamos cerrando.
Es solo para responderle a Paul y Adriana, que tienen razón, es un tema que ya se nos está viniendo encima. Hemos tenido reuniones entre todas las entidades, evidentemente el ICA ya avanzó bastante porque ya tiene su regulación, pero nosotros lo tenemos en nuestra agenda que esto tiene que salir antes de finalizar el año. Tenemos que tener, como Ministerio de Salud e INVIMA, tenemos que tener ya una regulación específica para el tema de salud humana, y es algo que está priorizado en la agenda en el tema de biotecnología.
Perfecto, muchas gracias. ¿Un comentario? Sí. Una acotación pequeñita.
Gracias. Yo los escucho hablar de los temas normativos en las otras áreas y sí les recomendamos pues a esos otros sectores, otras áreas, y cuando digo a esas áreas no es solo gobierno, sino instituciones de investigación, academia, gremios. Tenemos que vincularnos, articularnos para construir mesas de trabajo y no esperar solamente hasta que la autoridad tome la iniciativa quizás de hacerlo, sino tiene que ser también desde la base, y la muestra del resultado de esta norma que se tiene hoy para el área vegetal es la articulación interinstitucional y también una articulación con nuestros países para poder revisar modelos en otros y mirar cómo lo vamos validando. Pero es quién va a tomar el liderazgo y cómo se van a, o cómo nos vamos a sentar en esas mesas. Y es urgente.
Bueno, sin más, Food Safety, Seguridad Alimentaria nos llama, entonces daríamos por terminado esta sesión. Los esperamos después de almuerzo porque vienen en la tarde charlas relacionadas también con el tema de edición de genes, algunas cuestiones generales, ejemplos particulares, y vendría nuestro taller de cierre. Entonces, sin más, levantamos la sesión y nos vemos aquí a la 1:30, si mal no estoy.